All right, thank you for that. Okay,
No
so
problem.
sure. I am unable to
Excuse me,
see.
Professor, do the polls dictate attendance for the course?
Yes. The polls
Okay.
will give me all the answers that you've chosen and things like that. Yeah.
But if we show up a little bit late, it's okay?
I mean, yeah, it's fine, but just make sure that it doesn't affect your, ability to, do well on the polls, right? I'm not grading them per se, but they do count towards your participation, right? They're just polls. I'm not grading them. All right. So going back to macromolecules, let's get started, right? So at the very basic level, what we have is, everything is made up of matter, and we're no different. We're going to be made of matter as well. And you think about, the types of matter that is found in the universe, right? The solids, the liquids, the gases, and, so far there are about, I believe, 92 naturally occurring elements that have been found. And, there are others that have been artificially synthesized in the laboratory. We are comprised of many of them, right? Many of them are found within our body, some to not such a great degree, but that doesn't mean that they aren't important to us. Out of the 92, about 24 are naturally found, are required for the body's function and so on, and others can contaminate our body, right, and can result in disease, right? So the goal here today isn't to talk about, what are those elements that may cause disease. I will probably mention just an example or two just so that you may understand that. Now, you guys have all seen what an atom looks like, which is basically the unit for an element, right? There are 92 different types of atoms that are out there, and you know that they're made up of protons and electrons and neutrons, where the protons and neutrons are found within the nucleus, and electrons sort of circle around that, right? And so the protons are positively charged and electrons are negatively charged. And generally speaking, in a neutral atom, you're going to have, right, equal numbers of protons and electrons. And that is what prevents an atom from acquiring a charge. But we know that that's not always true, right? A lot of the atoms are very, very reactive, and so they end up acquiring a charge. So we're going to talk about that in a minute, but generally the charge arises from the sort of the electrons that are found in the outermost shell that we refer to as the valence shell, right? And so generally speaking, there must be 8 electrons within that shell, except for hydrogen that just has the first shell, so it should have just 2 in it. But so an atom strives to complete its outermost shell. Either it tries to have 2 or it tries to have, 8. And that is what eventually gives rise to the various ions that we will talk about. Now, in terms of what are the elements that are found within our body to an appreciable degree. So here is a chart, again, from your textbook, which shows you the major elements. These collectively make up 99% of your body weight. So that includes your proteins, your fats, your water weight, all of it, right? And majority of the body weight is comprised of, oxygen, right? 65% of your body weight is going to be comprised of oxygen, and then you have carbon and hydrogen and so on. But, the elements that are listed on the left end of the table, oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus, they make up 99% of your body weight. So we refer to them as major elements, right? And they make up our macromolecules that we'll talk about today, the proteins, the carbohydrates, the fats, cholesterol, nucleic acids and things like that. But of course, there are also other elements. Like I said, initially, you're going to have, about 24 elements that are naturally found within your body to perform various functions. So outside of the 99%, you have 1% of the body weight that is comprised of some other elements, such as sulfur, potassium, sodium, chlorine, magnesium, and iron. It's not important for us to talk about these minor elements individually today because they're going to come up during our discussion of physiology of, say, how muscles contract or neuron, electrical impulses are sent along axons of neurons. So we will get to them, right? But the important thing today is to just understand that they make up a very, very small portion of your total body weight. And sometimes they are found as part of, macromolecules. For example, you can find sulfur in, in proteins, not nucleic acids, is found in proteins, right? But majority of others, potassium, sodium, calcium, things like that, they're part of minerals, right? And they aren't very stable atoms, so they tend to either lose or gain electrons to become stable, and they're found actually in their ionic form, right? So that is how our body is able to really use them. If they are not in their ionic form, then they are of no use to us. Now, so if the ones that are listed here make up less than 1%, right? So there is still a very tiny portion, okay? So 0.02% of your body's weight is comprised of what we refer to as trace elements. So think things like zinc, manganese, right? Other nutrients that your body needs as minerals as well. And, they're extremely important as well, because as we're going to see in a week or two when we talk about enzymes, that enzymes cannot function without what we refer to as cofactors. So these trace elements help enzymes carry out their reactions. And if they are missing, enzymes cannot be functional. If you have them in a quantity greater than that trace amount, then they become toxic to you, right? So, but generally think of all of these minor elements and trace elements as helping, okay, the macromolecules carry out their functions. Yes, Purnima, you have a question.
What are trace elements again?
Trace elements are elements that are not listed on this table, but things like zinc and manganese, right, that are required for proper functioning of enzymes because they help act as a cofactor. Okay. All right.
Thank you.
Sure. And so, of course, even between when you look at these various minerals that are, or I shouldn't call them minerals right now. I mean, they're just listed as elements, but in the body, they will be found as part of a mineral, like calcium and phosphorus are found together as calcium phosphate in your bones, right? So you have, calcium that is found in the highest quantity, right? 1.5%, phosphorus, next, and so on. So, you're going to have, even between these minor and trace elements, those that are found to a much higher degree versus those that are not. Guys, if you have a burning question, you can unmute yourself and ask, okay? All right, so, what are some of the sort of places on the periodic table for these elements? And the only reasons why I'm showing this is because I want you to understand that, they kind of belong at certain places on the periodic table. And of course, I don't have time to describe the entire periodic table, but generally speaking, if you look at the periodic table and to your left or to the right of the periodic table, you have elements that tend to lose electrons, right? And you have the numbers on top, these Roman numerals that tell you how many electrons they would lose. So those in the first period would lose 1, those in the second period would lose, 2, and so on. And then you have the other end of the periodic table. And here, generally, you would have atoms that tend to gain electrons. So oxygen tends to gain 2, chloride tends to gain 1. These are what we refer to as noble gases. Their outermost shells are already complete, so they don't want any more electrons. They're inert, they are not reactive. But look at like oxygen and sulfur, for example, in the same period, both of them are going to end up, gaining 2 electrons, and acquire a negative charge in the process, right? So I'm kind of hoping that you remember that some of that information. So you should just be sort of brushing up of what you already know. And iron kind of is found in the middle, and it tends to generally lose electrons, and you would have iron ions that either lose 2 electrons or lose 3 electrons, and you can have ferric or ferrous ions, and that's how the iron is found within your body, okay? Now, the reason I talk about it is because, like I said initially, that these elements are going to be reacting, right? And they react to form various chemical compounds. So this video is gonna walk you through the 2 types of chemical bondings there are, because it will be much more rapid than me describing it, and then we can just get to what we need to talk about. So I hope you can hear it. If not, I'm gonna give you the link for the video and give you a couple of minutes to watch it. All right, so let's see if we can watch it and you can hear it. If not, just speak up.
I don't hear anything.
You don't hear anything?
Can't hear you, Professor.
Okay, so what I'm gonna do is I'm gonna stop. Okay, I'm gonna stop and I'm gonna give you a link for this in the chat. Okay, and you guys can watch it there. And then I'm gonna be back in a couple of minutes. Give me 1 second, figure this out. Okay. I'll just keep. It's kind of taking the same amount of time. All right, so I just put in the link for the video in the chat. Oh no, hold on. No direct messages to everyone. Hold on. Okay. So I'll give you a couple of minutes, watch it, be back here, okay? Are we done? Okay. All right, I'll give, until, yeah.
I'm actually pausing the video to take notes.
Okay, no, keep on watching it. I'll give another minute. That's fine. We'll be back at 9:45, everyone, okay?
Professor, where did we have to go? I didn't hear what you
There's a
said.
link. There's a link in the chat here. I'll put it in again. Okay, click on that, go watch it, and
And then what do we do after we watch it?
then you come back here.
Okay. Thank you.
Yeah, Carlissa, you should keep up with your readings. I am sticking to the syllabus, so there is that change from, doing chapter 1 and 2 for the first week to now chapter 1 for first week and chapter 2 for second week. So I have updated the syllabus for that. So look up the lecture schedule and stick to that. Please mute yourself. Mute yourself, please.
I'm kind of hearing everybody's thing and it's kind of annoying.
Mute yourself, please.
Professor Chima, I just had a quick question about ionic bonding.
Yes, go ahead.
It's been a long time since I took high school bio and chem, so forgive me if I'm missing something, but can molecules give up more than one electron? Like in the video, they give an example of just sharing the one. They can give up 2 though, if it is.
Right, so if we look at magnesium, so this is what I mentioned earlier on, so that the second period, magnesium and calcium would give up 2 electrons.
Okay.
It's a lot easier for them to give up 2 electrons to have a complete outermost shell rather than gaining 6, right? So it depends on what we can do. Yes, absolutely.
Thank you.
Excuse me, Professor?
Yes.
Yes, why is sodium chloride stable, but if it's sodium just itself, it's explosive?
Because sodium does not want to keep that 1 electron. It just makes it very, very unstable. But once it explodes, it has already given up its electron because it's reacting very, very quickly with oxygen. And so it's, it's going to be oxidized in the process. And after that, you're really, it has become stable. And that's how it is with sodium chloride, that as long as sodium chloride is in its solid form, it will be like a stable interaction between sodium and chloride. But once you dissolve it in water, it's going to disperse. That's an important concept that we're going to talk about next.
Okay, thank you, Professor.
All right, everybody, come back. Okay, we're going to start. So one of the first, so there are 2 types of bondings, right? There is the ionic bonding and there is the covalent bonding. And we'll talk about the ionic bonding first because it leads to production of ionic compounds, right? And so these tend to generally be minerals like, as was just mentioned, sodium chloride happens to be one. And of course, this is the table salt that we eat, right? So once the ionic bonds form, right, it's basically an attraction between oppositely charged ions, right? So in the example shown, the attraction was between the sodium, which has become positively charged, and chloride that has become negatively charged, right? So as long as they remain in the mineral form as like a dry table salt, they will be together as NaCl. But as soon as you dissolve it in water or you put it in your mouth and there's plenty of saliva and water in your mouth, they will dissolve in water and also disperse, all right, or dissociate. What that means is that they will kind of leave each other, right? And they will instead interact with water molecules. And we're going to talk about why they would interact with water molecules in a moment. Now, generally, when you have salts such as sodium chloride or acids such as HCl, or there might be other acids like sulfuric acid, H2SO4, or bases such as NaOH, all of these are going to disperse. So although HCl and NaOH, they aren't exactly held by ionic bonds. However, they kind of disperse just the same way that minerals such as salt do, right? So please do not think of them as being, chemical compounds that are ionic. They are, held together via covalent bonds, but acids by definition are compounds that give up a hydrogen ion or a proton as we refer to it, and bases by definition are the ones that either give up hydroxyl or accept a hydrogen ion, right? So, they, because of their the chemical nature, they're kind of shown as part of this image. I just want to point that out because I don't want you to make that mistake when you come back to this image and look at it and think that they are also ionic compounds. They're not. So salt is, right? And so when it dissolves in fluids, it tends to be surrounded by water molecules. So sodium has sort of separated from chloride, and two of them are individually surrounded by a bunch of water molecules. And so, if you've ever heard that water follows salt, right? What we're essentially saying is that once the sodium chloride has become dispersed, anywhere where sodium would go, it would take with it the hydration shell that surrounds it. Okay, so sodium is not only followed by water, it's actually also followed by chloride, right? Because the two are oppositely charged. They still have an attraction for each other. So wherever sodium is, that is where chloride would end up. That is where water would end up, okay? And water would follow all types of ions because that's just how these ions are found in bodies' fluids, right? So if you have a lot of sodium inside the cells, water would follow and end up on the inside of the cells, right? If you have a lot of sodium outside of the cell, the reverse would happen. The water would follow, and end up on the outside of the cell. So that's not the only reason water follows, but just kind of get you to start thinking about why water moves from one place to the other, right? And so you have a lot of sodium and chloride in one compartment of the cell. They need their hydration shells, all right? They need to be surrounded by water, so water follows, okay? All right, so just keep in mind these negatively charged ions are referred to as anions and positively charged referred to as cations.
Professor, are ionic bonds strong?
They're strong?
Are they strong? I'm asking.
Yeah, they are, they are strong as, like I said, within water, they will disperse. But if you were to remove water, right, if you were to make a solution of sodium chloride, right, and then you afterwards maybe evaporate the water by boiling it, you would again end up with the minerals. So yeah. Okay. Yes.
Professor, could you explain hydration shells again?
Hydration shells, and I guess I haven't talked about water yet, so it might not come intuitively, and I'll come back to that in a minute. But hydration shell, just think of it as water molecules surrounding ions. So it could be a proton, like a hydrogen ion. It could be hydroxyl ion. It could be magnesium. It could be calcium. In this case, you're seeing sodium and chloride, right? So any charged ion that you have, whether cation or anion, will be surrounded by water molecules that we refer to as a hydration shell. So notice the water molecules form like a shell around each ion, right? And so that's
Yeah.
what a hydration shell.
Thank you.
Sure. Right. So, you can, salt dissolves in water to give you, sodium chloride dissolves in water to give you one cation and one anion, but you can have other salts such as calcium chloride, which, dissolve in water to give you 1 calcium ion and 2 chloride ions. So, depending upon the mineral that you have, right, the amount of water that will follow would vary, right? So here you're just seeing 2 hydration shells with 1 sodium and 1 chloride, ions. But when you have 3 ions with 1 calcium and 2 chloride ions, there's going to be 3 hydration shells, right? So at least 1/3 more water that would follow that. Yes, Alex.
Okay, just last question. So it's okay in our bodies to have sodium if it's the cation, because that's not going to be harmful. That can eventually reform sodium chloride or whatever. But if it was sodium with 1 electron, it would be very devastating to our
Right,
bodies.
exactly. Okay, so I see, what you're asking. So within the body, right, sodium in its cationic form is the only form it's usable. Okay, it's not usable as a crystal, as a NaCl together, right? But once it dissolves, it forms what we refer to as electrolytes, right? So if you think of electricity, right? Electricity is basically flow of electrons or ions, and in this case, we have ions such as sodium and potassium and a bunch of others that will help to conduct that electrical impulse that travels along neurons, or that helps to contract, your skeletal muscles and so on, right? So they are only useful in their ionic form, right?
Would sodium be deadly if it was with the 1 electron?
Absolutely, it would, yeah. I mean, as soon as it comes in contact with the air, you can't keep it dry. Generally, it's kept submerged in some type of solvent so that it's stable. If you allow it to come into contact with oxygen, it kind of just burns away immediately. So it's not like you'd be able to put it in your mouth. Right, so yeah, it's very, very reactive, and you're never going to see it in its pure form. Okay. All right, so again, just to go back to, electrolytes. Electrolytes are basically ions within your body that would help to conduct electrical current. And we're not there yet. We're not ready to talk about the conduction of the electrical current. We'll come back to it. Today's purpose is to just kind of talk about the ions that help make that happen, right? So if you consider this image, perhaps a cell, right? So cell membrane in its resting state, it might not make sense to you right now. We'll come back to what resting state is. Just think of it as a cell membrane of either a neuron or a muscle cell. And cell membrane tends to separate the inside of the cell from outside. So inside being the cytoplasm, the outside being what we call the interstitial fluid or extracellular fluid, right? Fluids that are found outside of the cell. Okay, every cell within your body, regardless of the trillions of cells you have, it must be surrounded by a little tiny bit of water, because that is what's going to help transport, move things across, right? Across the plasma membrane. So think of this as the plasma membrane, the bar in the middle, and then you indicated is the outside of the cell or the interstitial fluids, and the inside of the cell where the cytosol is. And what I want you to notice is the difference in ion concentrations across the bilayer. So you're going to have a lot more potassium generally found within the cytosol, right? So if you were to take a sample of the interstitial fluids, which are outside of the cell, and compare the potassium ion concentration of the interstitial fluid with that of the cytosol, right? You're going to find a lot lesser in the interstitial fluid than inside the cell, right? And that is tightly maintained, all right? So there are homeostatic mechanisms. You'll talk about, ion balance towards the end of Bio 41, but it's tightly managed, right? Any change to that and your cells are unable to conduct the action potentials or the electrical impulses. So it's extremely important. You can't move your muscles, or, depending upon what the imbalance might be, you might have spasms in the muscles, which can be very, very painful. And because these ions are also important in conducting neuron impulses, and if the imbalance hits your nervous system, then you're unable to use your nervous system as well. People can go into coma because of the imbalance in these ions. So potassium is one that's important. Equally important is sodium, right? So notice the concentration. You have a lot more sodium outside in the interstitial fluids than you have in the cytosol. So it's opposite of that of potassium. All right, so potassium is more inside, sodium is more outside, and that is how it must be maintained. In the previous slide, I mentioned wherever you have sodium, that is where you will find chloride. So chloride is going to be a lot more in the interstitial fluids than you're going to find it on the inside. And of course, there are many others, right? So today we'll talk about a bicarbonate ion that is going to be mostly found in interstitial fluids in your blood plasma because it helps to maintain blood pH. And you would have other ions such as phosphate that are found in the cytosol. And why am I mentioning these negatively charged ions is because I want you to notice that there is both cationic and anionic species that are found both on the inside and outside of the cell, right? You don't want to separate the charges too much because that can again be destructive for physiology, okay? So a couple of things, right? So I said that wherever you have sodium, that is where majority of the water would be, right? So if you consider this to be a resting state, a balanced state, if any imbalance were to occur, so that there was going to be a lot more sodium somehow, it ended up on the inside, then water is going to follow that. Okay, so there is the electrical effect of these electrolytes. Electrolytes are any ions, any minerals, acids, bases that disperse and give rise to these types of ionic species. Okay, so anything that has a charge can potentially be an electrolyte. But as I mentioned earlier, that there are certain ones that are found in your body to a much greater degree than others. Sodium and potassium and chloride are going to be much more important than, say, magnesium in conducting the electrical effect. So when we're talking about how nerves conduct their business and how muscles contract, we're going to come back to sodium and potassium and chloride and things like that. Okay, so they're extremely important in helping your central nervous system, your control center, remain in control of your body. And then, of course, I just mentioned the osmotic effect. Osmosis just refers to the fact that water can move across the bilayer, easily. Okay, so it's not hindered. Plasma membrane generally tends to be semi-permeable, right? It controls what goes in and out. Movement of sodium and potassium and chloride and all of these ions is very tightly regulated. It's allowed to enter at times. It's prevented from entering at other times, and we're going to talk about transport, next week. But if you consider water transport, it happens unhindered. You can't control it, right? It just, every cell has no sort of ability to control whether water moves in or out. Okay, so some of you have already talked about diffusion and osmosis in lab. Those of you who haven't, you'll talk about it today. If not, I'll come back to it in lecture and I'll talk about it. Right, so certainly wherever you have more electrolytes, right, that is where water would be. So water follows electrolytes, right? So this is why if you've been, exercising a lot and you've lost a lot of electrolytes because you've been sweating a lot, you're advised to drink, some electrolyte-filled fluids, right? And the reason is that if you take in salts, the water is much more likely to be absorbed into your body than if you were to just plainly take water, okay? All right, so it controls the water content and distribution within the body. And then, of course, these ions aren't just there for the electrical effect and osmotic effect. I mentioned one thing already, that things like manganese and zinc are important in carrying out chemical reactions. And, things like phosphate and things like calcium, right? They are part of the structure of the bones. If you don't have enough calcium and phosphate in your diet, you can have very, soft bones, okay? Which we'll come back and we'll talk about when we talk about the bone physiology. I don't want to take too much time going off on tangents right now. So, they're extremely important in chemical reactivity as well, okay? So, the diet that you take, as long as you take a normal diet, you're taking in a normal amount of these electrolytes. You don't need to be drinking and a lot of fluids that have high content of electrolytes. That's generally for people who work out a lot, right? And they lose a lot more electrolytes on a daily basis than somebody like me who doesn't work out, doesn't exercise that much as compared to somebody who's an athlete, right? Right. So it's extremely important, as I said, that you must have adequate amounts of these ions in your diet. You can't have too much of it because that's bad, and you can't have too little of it because that's bad. And generally, your body has homeostatic mechanisms that maintain it. And if you've gone to a point where you have an imbalance, then there has been something going on with you for a lengthy period of time. That's Alex.
Yeah, Professor, why do athletes have such a low resting heart rate? Does it have anything to do with electrolytes?
They have a low resting heart rate because they generally have conditioned themselves. And generally, when we start exercising, right, we will, like, let's say me, who's not an athlete, if I start exercising, my blood pressure, my heart rate initially goes up because of all the work the muscles are doing, right? I begin to breathe a lot more heavily. And the reason is that, my muscles aren't conditioned for that type of activity. They want a lot more oxygen. They don't store that oxygen that they need once they start exercising. They don't store the nutrients that they need once they start exercising. And your neural circuit that controls the heart rate, right, is not at the same, essentially. Okay, just to put it in simple terms, you will talk about it when you talk about the cardiovascular system, but it's not exactly the same as that of an athlete. All right, so for somebody who's an athlete, their muscles are storing much more glycogen, right? This is why they eat a lot of carbohydrates, right? They'll eat like a big pasta meal or whatever. They they lose a lot of electrolytes, so they kind of continue to replenish it. There are a lot more blood vessels that are reaching their muscles, and they're providing it with the nutrients and oxygen and taking away the carbon dioxide at a much rapid level. So there are multiple things that are different between an athlete and a non-athlete, right? So when we say that an athlete, their body's been conditioned for a particular activity, over time, their body has been adapting, right? So their homeostatic mechanism when it comes to heart rate, right, has changed from my homeostatic mechanism, right? My body's clue is to, once I start exercising, the response is that it, increase the heart rate, increase the breathing, increase the blood flow to all the muscles. But, with an athlete, right, this is just how their body has been functioning for long periods of time. So they don't need to, their body doesn't require that, right? There is already a lot of nutrients there. There's already enough oxygen being sent there. So they don't need to speed up any of that process. And so their heart rate remains low.
Professor, how long would it take you to get to that level? Would it take like 2 years of exercise every day?
I think so it depends on individuals too. It depends on your body. You would have to be continually exercising, on a daily basis, eating right. It can take some time. I'm not sure the exact amount of time.
Yeah. It takes like a few months, honestly. If you exercise 3 or 4 times a week, your resting heart rate will go down. Yeah, it's like 2-3 months.
Okay, thank you for that. Yeah, so it does drop. I can tell you that if I haven't been exercising for some time, and then I go back to it after, say, 2-3 months, again, I start to breathe heavy and all of that. But, if I continue to do it for a week or two, things start calming down, right? My blood pressure isn't that high anymore. My heart rate isn't going crazy. So it can happen rapidly. Okay. All right. So, okay. So again, you know, this table isn't for you to memorize things just yet. We will come back to it every time we talk about these ions and, you know, their involvement in physiology. But a couple of things that I want you to remember, and I think I've kind of shown you on this image, is where are they mostly prevalent? It isn't to say that they aren't found inside the cell. If you have majority of the ions outside of the cell, they're found both inside and outside, but their role is mostly to be in the compartment where they're where their presence is heavier. Potassium, you just saw that again, you know, a lot more presence inside within the cytoplasm than outside. You know, both of them are extremely important in muscle contraction and neuron physiology and making your brain function and things like that. So we'll talk about that. In fact, one of the first things we'll talk about is gradients, which I just mentioned to you. But again, we'll come back to that later. Calcium, I mentioned, you know, found in bones, but it's also extremely important outside of that. It might take part in, you know, chemical reactions such as the clotting, the neurotransmitters, which must be released from neurons so that, you know, a muscle could contract, for example, or another neuron might be stimulated. You know, it can't happen without calcium, right? So you must keep, you know, your calcium levels high. If you don't, then your bones begin to dissolve to release all of that calcium. So just to give you a couple of examples. And then chloride ion, it's not just in your body to kind of follow around sodium and kind of neutralize the charge. That's not what it's there for only. We're going to see that it's involved in preventing the neurons from firing an action potential, right? And which is equally important, right? Just like muscle contraction is important, it's, you know, important to relax a muscle as well, right? So your homeostatic mechanisms are comprised of opposing actions. You contract, you relax, right? You fire, you want to stop firing and things like that. So we're going to see chloride has an important role in that. It also has a very, very important role in exchange of gases, okay? So whether it's across individual cell membranes or if it's across your lung surface, it's without that chloride, you would not be able to exchange carbon dioxide for oxygen. Your stomach would not be able to produce the acidity needed for you to digest food. So it's extremely important as well, okay? And so you'll talk about things like, you know, chloride shift in erythrocytes, which is essentially, again, just exchanging the gases, okay? Bicarbonate ion, we'll talk about today, which is part of you know, the pH balance within the body. So we'll get to that. And phosphate ion, which is extremely important because it's part of ATP, which we consider the cellular currency. You know, without it, the enzymatic reactions can't occur at the rapid rate that they do within our cells. And it's also part of, you know, important macromolecules, the RNA and the DNA, which is, you know, your genetic material and copy of the genetic material. You know, the phosphate ion is an integral part of that macromolecule. So we'll talk about that as well. So while chloride happens to be the most abundant extracellular anion, phosphate happens to be the most abundant intracellular anion, as I showed you on the previous image. Right, so let's see. You guys read the question. You guys have, let's come back at 10:13, okay?
Professor, can I switch devices while we're on break?
Yeah, of course.
Thank you.
Hey, Professor, can I ask you a quick question?
Of course.
So I was just wondering, what happens if you have too many electrons? Not electrolytes.
Yeah,
Sorry.
so too many is bad too. So like you might have heard of things like hypokalemia, which is too little potassium, and hyperkalemia, which is too much of calcium. So they're equally bad. Okay, they will impair your nervous system function. They will impair your muscular function. They will impair your digestive system function. They'll impair all sorts of function. And if it's not taken care of, it can be very, very dangerous and can result in death over time.
Jeez. Okay. Wow. Sorry.
I know it's something that you don't want to play around with.
Professor, can electrolyte imbalances cause like muscle twitches?
Yes, it does, because like I said, sodium and potassium are both important in maintaining, in allowing your body to contract your muscles and relax your muscles. Okay, so it's not just, remember, homeostatic mechanisms are opposing functions, right? So if you contract, you want to be able to relax. And if you have twitching, that's certainly pointing to imbalance, because your body is doing it, if it's skeletal muscle, for example, your body is doing it without, your intentional input, without you actually wanting to do it.
Do we answer the question in the chat, or we just wait for you to come back?
Yeah, we'll come back and give it another minute so everybody has a time to think about it, and then I'll just ask, and we'll just go from there. So you guys have some sort of a response ready?
You said that chloride is the most common extracellular anion.
Yes.
And what was the most common intracellular?
Phosphate.
Okay, thank you.
All right, so who has a response for this? Raise your hand so that I could call you. You don't have to have a complete response, but I would like for you guys to maybe first describe what's going on with Kelly. Okay, so let's see who's raised. Rebecca.
Is it because she has low electrolytes?
Yeah, and what happened? Why are they low?
Because she sweated them out.
Right. So when you are sweating, you're not just losing water, you're also losing a lot of electrolytes, right? So that's probably one of a major way you can lose electrolytes. So regardless of whether you're running, exercising, it's a hot day, you're gonna sweat a lot, you're gonna lose a lot of electrolytes as well. So, Darshan, what should she do, or what has she done wrong first?
Well, I think that because she did like such a big run right before her actual race, and I think that was the first step. You should rest right before you're doing any big competition. And also she didn't well, it just seems, according to the question, it seemed like she just drank water and went to sleep. That's also something that she did wrong, because yeah, it's good to drink water, but like also you have to fuel your body, like maybe like Gatorade or even like carbohydrates. She's not refueling anything. She just drank water and went to bed. So
very good. Awesome observation. All right, so Tasneem, what what's going on the day of the race, of the marathon?
Oh, Professor, I actually had a question regarding the muscle weakness. So I was wondering if a potassium and a calcium deficiency can cause cramps.
Yes, absolutely. Yes.
Oh.
Yeah.
Because that happened to me. That happened to me one time while I was doing like a 100-meter sprint, and I realized my thighs and my calf muscles were like contracting like crazy. So, and my PI instructor told me like
exactly what it is. So it's a potassium deficiency, right? So drink electrolyte filled fluids like Gatorade was mentioned. Eat bananas, right? I actually get those a lot. So the way to get rid of them is if you wiggle your toes, right? That kind of is using muscles, and obviously your calf muscle is right now in a spasm state or whatever. But yeah, that kind of helps to soften it up a little bit, and that goes away, the cramp goes away. But yeah, it's a potassium deficiency. Bananas have a lot of potassium. Eat a couple of bananas and you should be okay.
I see. Thank you.
Sure. Yeah. So what is so I guess that answers the question, right? So that's what she's feeling, that the weakness and cramps is coming from that loss of electrolytes, and she should have replenished them by not just drinking, water that's needed, but she also needs nutrients so that her muscles can again stock up on the nutrients. So when she starts running, they're there. And of course, none of the neural function or muscular function is possible without the electrolytes. So, that's the imbalance that she's feeling. Any questions? I know some of you raised your hands. Allison, Autumn.
Yeah, just one quick thing that I learned very recently is that Gatorade actually has like less than 1% of the recommended daily intake of potassium specifically. So it's very common to think of Gatorade as like a good thing to replenish your electrolytes, but they're just, so everybody knows, it's not really that great for electrolytes.
Thank you so much for sharing that. But again, it might be important for somebody who's an athlete, right? But not for somebody like me, right? I probably have enough potassium in my diet, and that Gatorade might be sufficient.
Professor, what drink is good then, though? If Gatorade's not
I
good?
wouldn't know. I'm not comparing.
Can that same girl come back and answer the question?
So there's what I do, because I do long distance running, I use a magnesium supplement that's a powdered form. It's like a flavored powdered form. And then what I do is I use it's called like light salt. It's designed for people who have high blood pressure, but it's actually half potassium and half sodium. So if you mix those two, then you're getting the magnesium, the sodium, and the potassium all together. Obviously, I am not a doctor. Don't take my advice. And
that's exactly what I was going to say.
Right, but please do your own research and consult your doctor about.
That sounds like a lot of work to put all.
And there should be like a workouts over an hour.
Yeah, for you to be actually taking in a lot of supplements and things like that. Yeah, but definitely, it's good to have this information, but it's terrible to apply that.
Just consult your doctors, guys. Sorry.
Exactly. No, absolutely. I agree. I think that's extremely important because, this imbalance, it creeps up on you, right? It's not like your body is going to give you all of these signals, although like muscle cramp is a signal, but that's a normal thing, right? It's your body beginning to tell you, hey, you're running low. But once you're really in a dangerous category, you might not remain active enough to realize that. So you don't want to be playing around with that. All right.
Pedialyte be?
Say that again.
I'm sorry, I don't know like how to ask questions and not interrupt someone, but how helpful would Pedialyte be?
So again, I believe Pedialyte the ion concentration might be low because it's generally the concentration recommended for children, right? But you can again check with your doctor and see what's best for you. Okay. Yeah, don't just start playing around with yourself. All right. Okay, let's move on. Now, the second type of chemical compounds that are just as important are the ones that are where atoms are connected together via covalent bonds. And again, in the video, it showed you that it's the sharing of the electrons, right? And so generally the macromolecules that we're going to discuss next are held together via covalent bonds. So things like carbohydrates, fats, nucleic acids and proteins are all held together via covalent bonds. And generally, at the heart of the chemical compounds that make us up, the macromolecules that make us up, is carbon, right? Carbon and hydrogen. We're made up of macromolecules that are, heavy in carbons and hydrogen. And of course, we're going to see with the case of carbohydrates, a lot of oxygen. With the case of proteins, in addition to oxygen, you're going to have a lot of nitrogen. You're going to have, some sulfur. In the case of nucleic acids, you can add nitrogen and phosphorus to that as well. So, structurally, they're going to vary a little bit, but just keep in mind the hydrogens can only form a single covalent bond. Oxygen can form double covalent bonds, and nitrogen can form triple covalent bonds, whereas carbon can form, quadruple. So they can form 4 covalent bonds. And as you can see, the carbon atoms here, if we consider just the first one here, that where my cursor is at, it's bonded to 1 hydrogen, 2nd hydrogen, 3rd hydrogen, and the 4th bond is between the carbons. So, The
Just
idea.
repeat that, Professor, please. Hydrogen form how many bonds? Can you repeat that, please?
Sure. So if you look at this image, notice here, there is sharing of a single pair of electrons. So there is a single bond, covalent bond between hydrogen atoms, or any hydrogen, regardless of who it's covalently bonded to, it can only form one hydrogen, one covalent bond, okay? And oxygen, notice there are 4 electrons that are shared. So that's 2 pairs. And so that's a double bond, okay? So oxygen can form up to 2 covalent bonds. In this case, both bonds are between oxygen molecules, but they don't have, oxygen atoms rather, but they don't have to, right? Oxygen can form a bond with like 2 separate hydrogen atoms to give you water molecule, for example. Here in this case, there are 3 pairs of electrons that are being shared, and so nitrogen is able to form 3 covalent bonds. In this case, we're seeing, molecular nitrogen, where you have, both all the covalent bonds between those 2 atoms, and so that's a triple covalent bond. But again, as was the case with oxygen, this nitrogen can form those 3 bonds with individual atoms. So ammonia, which is NH3, nitrogen is bonded to 3 separate hydrogen atoms. And then you have carbon, which can form 4 covalent bonds. And notice here, oxygen has formed those 2 covalent bonds, one with hydrogen and the other with carbon. So the goal here is that just keep in mind that because of their ability to form these tremendous number of covalent bonds with each other or with other atoms, they can give rise to a diverse, complex, a series of chemical compounds, right? And that is what makes, all of the macromolecules and the diversity that we find within our body possible, right? They can be in chains, they can be branched, they can be circles, and that's what you're going to see. And I'm not asking that you figure out how to make these structural formulas and figure out how atoms are, a part of a molecule, but just because it's important for your understanding of why macromolecules, for example, don't break down and make electrolytes, right? Why isn't glucose an electrolyte, right? Because it's not formed of, ionic bonds, and, it doesn't dissociate the same way that sodium chloride does in the body. So glucose is not going to form electrolytes, right? It's used as a nutrient to burn for ATP, and then that's what will fuel the muscles, right? But they are not going to form electrolytes, and so keep in mind that anytime you have these covalent bonds, it's equal sharing of electrons. It's not a give and take between atoms, and so there isn't going to be ions that are forming. So if you were to dissolve these ionic compounds in water, they are not going to disperse the same way that sodium and chloride does. Okay, so the diversity of carbon compounds is quite complex, and we're not going to talk about all of that. Of course, the carbon compounds are referred to as organic compounds, and there is a very difficult course associated with the chemistry of organic compounds. So just know that, macromolecules are organic compounds, and generally they are covalent compounds, right? That, the atoms are actually being shared, and electrons are not being donated or received. And so these covalent compounds do not disperse and do not become electrolytes. Okay, let's take our first poll. Okay, and it's based off of things that we've just talked about. So I'm gonna give you, how many questions are there? 1, 2, 3. All right, let's take 5 minutes. Poll is up right now. I don't know why you are not able to access it. Are you using a computer or a phone? Don't use an app. We still have polls coming up, so you should be able to see it. Maybe minimize the window. It might be somewhere in the back. I'm not sure. I have no clue why you're not seeing it. All right, guys, ending in 5, 4, 3, 2, 1. All right, I'm just gonna give last couple of seconds because I see a lot of you still submitting. Okay, end poll. All right, so one thing, if somebody could point out why some students aren't able to see, so I know you can't tell me that, but how does the poll just pop up on your window when I make it available?
Yeah, it just pops up.
Yeah, so
Yeah, it just pops up.
yeah, so it might be just, you know, just minimize the screen, or if you want to just log out and log back in, because there will be further polls coming up. So maybe try that. Okay. I don't know. Okay. All right. So let's quickly share the results and see how we're doing. Which of the following is a trace element? Okay. Manganese is the trace element. All right. If you look at nitrogen, potassium, and iron, you go back to the table that I showed you on slide 2, I believe, you know, nitrogen, you know, potassium, they happen to be part of the major elements. Iron happens to be part of the minor elements. And the only one that isn't even part of that list is manganese. So that is going to be a trace element. Which of the following will form a negative ion or an ion, right? And so there are a couple of responses to this. So yes, certainly iodine is one and oxygen is the other, because it tends to accept, you know, ions if it was part of that type of a reaction. Magnesium and sodium are towards the other end of the periodic table, and they tend to lose electrons, so they form cations or positively charged ions. Okay, which of the following doesn't disperse in water? Who wants to take that? It is CO2, and why is that? Any ideas?
It's not polar.
And also isn't CO2
Covalent bond.
released form of glucose?
So carbon dioxide, think about the types of bonds, you
Has
know.
the double bond, no?
Yeah, it's a covalent bond.
Double bonded and
Double
they're
bonded.
collab.
I mean, I just thought like there's bubbles.
Yeah,
in
so those are
soda, and there's
covalent
bubbles in
bonds that are
CO2.
holding CO2, and it's a very stable molecule. And if you look at the other examples I've given you table salt, which is sodium chloride. H3PO4 is carbonic acid, right? It has 3 hydrogens and a phosphate. It's an acid, and so acid by definition gives up a proton, right, or a hydrogen. So it will break down in water. This was one of the things that I talked about on that slide. So if you have acids, bases, or salts, they will disperse. Okay. If I had glucose here, for example, it would not break down into, it will dissolve, but it would not break down into like ions. It won't give you like, you know, charged oxygens and charged, you know, carbons and hydrogens and things like that. So only carbon dioxide. All right. Which of the following is true regarding the specified ions presence in your body? Okay, so the only one that's correct is that chloride is mostly found outside of the cell. Okay, sodium is mostly outside, right? Potassium is mostly inside. Calcium is mostly outside. So that's again that image that I showed you with the outside of the cell and the inside of the cell and the relative distribution of the ions across that bilayer. How many? Okay, so this I know it might not be the easiest question, but I just wanted you to think about it. I want you to think of, you know, making structural formulas and so on, but I just wanted you to think that there were 2 carbon atoms and they were just covalently bonded to 4 hydrogens, right? And together those 2 carbons should be able to form, you know, 4 bonds each, right? So there must be sharing of electrons more than one pair between carbon, right? So there is essentially, is there a double bond or is there a triple bond between them? Can somebody take that away? I mean, there's some who have said 1, the others have said, 2, 3, and 4. Who wants to take it away? Who has raised their hand? Yes, Denisha.
I was raising my hand regarding question 4.
Okay.
I'm a little bit confused. It says calcium is mostly found inside, and you said that's not the correct answer. It's chloride.
Right.
Could you explain that a little bit more? Because I think in my head I say calcium is inside the body because calcium is within the bones, correct?
So it's inside the body, but when I'm asking you these questions, I'm asking you to think at a cellular level. So if calcium is found inside the cell, it generally reacts immediately to form a salt, and it would become hard. Okay, that's how people get atherosclerosis, right? There is a lot of calcium that is taken up by their blood vessel walls, and they become hardened, and their blood pressure goes through the roof and things like that. So it's generally found in body's fluid. Most of what's needed in ionic form is found in body's fluids, and it's very tightly maintained, as we'll learn later.
Okay, I got it. Thank you.
Sure. Who was going to do number 5? Somebody had raised their hand, so if you could. Guest, so there's no name to you, so please go ahead and anyone?
Double bond?
Right, so there's double bond that is found between the 2 carbons, right? So each carbon is bonded to each other once and then to 2 hydrogens each, and then that leaves with just 1 more covalent bond, so that must be between the carbons because there's no more hydrogens to do that, okay? All right, so we will move on.
Professor, I'm confused. So the answer for number 5 is 2 or 4?
2. So the question is asking you, if you look at the question is asking you how many bonds between carbons, okay? So just the carbons. It's not asking how many bonds a carbon atom can make. If it was how many bonds a single carbon atom can make, it would be 4 bonds, but I'm asking you how many bonds are holding the 2 carbon atoms together, okay?
Oh, okay, sorry.
No problem.
Professor, quick question.
So the actual answer is it 2 or 4?
2, because it's just the carbon she was saying that's connected. She's not really asking about the hydrogen.
Okay, all right, I got it. Okay.
No problem. Thanks, guys. All right, let's move on.
Professor.
Yeah, go ahead.
My mic was not working when you were reviewing 1 through 4. Could you repeat the answers for 1 through 4?
I stopped sharing. So can somebody else, if you have the answers around, share it with him in the chat, please? Sure. Thank you. All right. So let's move on. All right. So covalent bonds are, important because they hold the molecular molecules together. But, the sharing of electrons isn't always going to be equal, right? So this is extremely important because that's important for the physiology of water, and we're mostly made up of water, right? The chemical properties, I should say, of water. And then, of course, 70% of our body can be water. So it's important, just as important for our physiology. So that's why I use that term. When we think of covalent bonds, we think equal sharing, but that's not necessarily true. There are covalent bonds where electrons are shared equally. Like if you have atoms of the same type, the sharing of electrons would be equal. They will spend some time around each of the hydrogen atoms in this case, and both will be happy. Now, in some cases, we will have unequal sharing of electrons. So chlorine atom, in this case, HCl, what you're seeing here, chlorine tends to pull the electrons with much higher affinity. Okay, it has multiple reasons, but one of the reasons is that it has a lot more, protons, so it can pull that electron with much more affinity than a hydrogen can. So the chloride, in this case, a chlorine atom, has a much greater affinity for these electrons. It wants it much more badly than the hydrogen atom. So we tend to call such type of atoms as electronegative atoms. Okay, so those that pull electrons with much greater force while in a covalent relationship with another atom, we tend to call them electronegative. And so if we were to look at the periodic table, you know, the electronegativity, notice it says increase in electronegativity. So if we were to just compare chlorine with fluorine, fluorine has the highest electronegativity, and, you know, bromine has much lesser than chlorine, and so on. So, you know, you can come back to the table and you can look at exact numbers that tell you, you know, the electronegativities of each atom. And so you can compare any bond that you wanted to, but that's not the goal here. We're not learning chemistry in that much detail. The whole goal is that you understand that in the macromolecules, we generally have carbons, you know, oxygen, nitrogen, you know, and hydrogens that are found within macromolecules. So we want to compare them in their electronegativities and see if a bond between, you know, carbon and hydrogen is that sharing of electrons the same as a bond between, say, you know, nitrogen and carbon, or, you know, oxygen and hydrogen. So to do that, you need to know the electronegativity. So I would kind of commit this to memory. When it comes to sharing of electrons, you can think of carbon and hydrogen at about the same level, right? So if you see any carbon-hydrogen bond, you can think of them as equal sharing in what we call non-polar covalent bonds, okay? And so, you know, any carbon hydrogens that you see, you know, non-polar, okay? What that means is that there is, think of polar and non-polar and ionic and covalent on a spectrum, where ionic bond is on one end of a spectrum and covalent bond is on the other end of the spectrum. And all of these other, you know, slight unequal sharing of electrons, what we refer to as as polar bonds rather, it falls somewhere between that range of covalent and ionic. Okay, so the nature of this polar covalent bond varies between the atoms that are involved. So again, I'm not interested that you memorize, you know, who does what, just so that you understand that, you know, you have the polar covalent bonds where there's unequal sharing of electrons. And how do you figure out if there is a polar covalent bond involved is by comparing the atoms that are involved. And for the macromolecules, we're interested in carbons and nitrogen, oxygen, and hydrogen. Let's compare them to the most electronegative element there is, which is fluorine, because fluorine has the greatest affinity for electrons. It takes part in any chemical reaction, it's going to take away the electrons. That's how it ends up, because it has the greatest affinity for them. Then comes oxygen. All right, so we're going to compare just the ones that we're interested in learning about. So oxygen has a greater affinity for electrons than nitrogen. So if you have nitrogen and oxygen part of a covalent bond, oxygen would pull electrons with a little greater affinity than nitrogen. And what that ends up doing is that it ends up giving oxygen a slight negative charge, and nitrogen a slight positive charge. Okay, and then you compare nitrogen with carbon, and of course, you know, nitrogen is more electronegative than carbon, and so, you know, in a bond between carbon and nitrogen, nitrogen is going to be slightly negative and carbon is going to be slightly positive. In a bond between carbon and oxygen, oxygen is going to be, you know, slightly negative and carbon is going to be slightly positive. Right? So the reason you want to look at this is because when you're looking at a macromolecule structure and you're looking at, you know, covalent bonds that make it up, you want to know if it will dissolve in water or not, right? So any compound that has oxygens and nitrogens and things like that, you know, they tend to dissolve well in water. And anything that just has carbons and hydrogens, notice hydrogen isn't even here because although it's slightly less electronegative than hydrogen, but that difference isn't enough to make it into a polar bond. Okay, so polar molecule will have oxygen and nitrogen and chlorine and fluorine and things like that. You know, they tend to be hydrophilic, which means that they are water loving, that they dissolve well in water. Non-polar compounds where there is equal sharing of electrons tend to be hydrophobic, which means they're water fearing. They don't dissolve well in water. Instead, they kind of form like a layer of oil over water, and they kind of are excluded by water, okay? And then there are molecules that we refer to as amphipathic molecules that will have both hydrophilic and hydrophobic properties, right? So now we're talking about large macromolecules like a phospholipid or a carbohydrate like glycogen, right? Or a large protein. So we're talking not at subunit level, at small individual, covalent compounds, but rather large macromolecules, right? So amphipathic just means that a portion of this molecule is hydrophilic and a portion is hydrophobic, okay? And that is important. That is extremely important in physiology. If we think about the plasma membrane, it's made up of amphipathic phospholipids that we will come back to later on, right? So just again, at the bottom of the image, you have a couple of examples of what a polar covalent bond versus what a non-polar covalent bond might be. You're looking at methane, for example, in the middle, carbon and hydrogen bonds. There isn't much of an electronegativity difference, so they happen to be non-polar covalent bonds. So that means that it's also hydrophobic. Methane is not going to dissolve well in water. Anytime you have a molecular oxygen or any type of those gases, they tend to also be non-polar, right? Anytime you have two atoms, they're going to share the electrons equally. Okay, and so anytime you think gases, think carbon dioxide as well, think oxygen, nitrogen, they don't dissolve well in water, okay, because they are non-polar. The only thing that kind of keeps them suspended in blood is the fact that the blood is continuing to flow and that they are carried by certain proteins, right? Oxygen and carbon dioxide are carried by hemoglobin, right? That's how majority of the oxygen and carbon dioxide is flowing through your body. It's not flowing as gases like bubbles dissolved in your blood vessels. That's not how it works, right? So anytime you think gases, unless they are either converted into another form so that they can dissolve well in water, which we're going to talk about in a minute with the carbonic acid, or that they are bound to a carrier protein, they will not dissolve well, right? So they will kind of form these pockets of air that can be very, very painful. Okay, and then you have a polar compound. Here you're seeing H2O, which is water. And in this case, you have the bond between hydrogen and oxygen is going to be a lot more electronegative than hydrogen. So oxygen is going to be slightly negative, whereas hydrogen is going to be slightly positive, right? So there are these slight charges that would allow something like sodium to be surrounded by water molecules, right? So sodium is positively charged, the negatively charged oxygen would surround it. Chloride is negatively charged, the positively charged hydrogens would surround it, right? So this polar nature of water makes it a great, great solvent. Okay, now this brings us to water. So we're just talking about water, right? So here again is the structural formula for water. Oxygen needs 2 more electrons to complete its outermost shell, and it's sharing that those electrons with hydrogens. Okay, so right now if you count, there are 8 total electrons. But because oxygen is more electronegative, these electrons spend more time circling around oxygen than they do around hydrogen. So that's why over time, the oxygen atom would be slightly negative, and it doesn't have 1 slight negative charge, it has 2 slight negative charges because of that sharing of electrons with 2 individual hydrogen atoms. And each individual hydrogen atom has a slight positive charge because it's only sharing 1 covalent bond with the oxygen molecule. So why is that important? This results in a molecule that has these charged ends that it can use to attract other water molecules in the area. It can attract ions, like I mentioned earlier, but it's also important to attract other water molecules. If you look at other molecular compounds that are similar in size or similar to water, they tend to be gases, like ammonia tends to be a gas. The reason water is actually fluid is because of its polar nature and its pull for, other water molecules in the vicinity. So remember, there are 4 slight charges on a water molecule, 2 slight negatives and 2 slight positive charges. So it's gonna make, bonds with at least 4 neighboring water molecules, right? So the slight positive charges on the hydrogens would attract the slightly negative charges on the water molecules and vice versa, right? So it's extremely important for our physiology as well. So this property of where water molecules are attracted to one another is called cohesion, right? This is what keeps the droplet on a leaf together, right? This is what causes, a tear to form, right? And then, or if you were to just spill a little bit of water on a countertop, the water tends to get together into a puddle rather than dance around. If you were to do like liquid nitrogen, for example, right, it would kind of, it would kind of dance around. If they don't have any attraction for one another, CO2 does the same thing and it kind of evaporates very, very quickly. Water doesn't do that, right? So, the reason it doesn't do it is because of its polar nature and its attraction for the neighboring water molecule that we refer to as cohesion, all right? They're cohesive, they attract one another. And then if you were to consider tear and the way tear kind of runs down your cheek, right? Or the way blood runs through your blood vessels, or how water rises up to the top of the trees via capillary action, right? That is something that we refer to as the adhesive property of water. So what water molecules are doing is that they're not only attracting to one another, they're also attracting to anything else that happens to be polar in nature, right? So they adhere to surfaces, right? So tear flowing down your cheek, there's a streak of water that forms because you have that adhesion of that water molecule for your skin, right? So both of them are possible because of the polar nature of water. And of course, water is extremely important, as I said earlier, that 70% of our body, adult body, is made up of water. So majority of it is found in the cytosol. And when you compare the various organ systems, the softer the organ system, generally is going to have a lot more fluids in it, right? Joints can have, a lot of lubricating fluid within them. So they're going to have a lot of fluids. Lymph is essentially, again, filtered plasma. So it's going to have a lot of fluid. So depending upon the tissue that you're looking at, the water content would vary, but some of the softer organs, they're going to have a lot more water than some of the harder organs, such as bones that have just 22 versus brain that has 75% water, right? And water percentage also changes with time, right? If you look at the skin, baby skin, that's, a lot more softer than, somebody who's elderly, because as you age, the overall water content begins to lower in your body. And that's why, elderly can heat up very, very easily too, coupled with the fact that their sweat glands might not be working optimally, but it's extremely important. So why is it important for us? So one, of course, it's going to help us dissolve like our electrolytes, right? I told you, like, sodium chloride in its mineral form is of no use to your body, but it dissolved as sodium chloride ions is extremely usable. So it helps to dissolve things because nutrients are only able to go across the plasma membrane if they are dissolved. If they aren't dissolved in water, things are going to stay wherever they are. Gases, nutrients, waste products, think of anything, it must be dissolved in water for it to be able to enter the cells or exit the cells. Water is going to take part in reactions. So generally, when we think about nutrient storage, we think about fats, or you think about, glycogen, they will not be broken down unless water was around. So it's extremely important to break down, nutrients within your body as well. We're going to see that it's found as part of, this lubricating fluid, right? So you've already talked about serous membranes and parietal and visceral and this potential space between them that has this lubricating fluid, and it's mostly water. right? So of course it has dissolved in it, other things, proteins and, ions and things like that. But without water, they can just harden up, right? And they're gonna not prevent that friction, which is the entire purpose of that serous fluid. And we're gonna see that, our organs, such as your brain, is fully sort of immersed in water, right? And every time you move, right, because the brain is kind of suspended in water, the water slows down the force with which the brain would move within your cranial cavity, right? If you swim, the resistance that you have to overcome to be able to pass through water. So brain would have to kind of do the same thing if it were to hit the one side of your cranium, which can be devastating. So one important feature that we're going to talk about today is thermoregulation. So we'll continue to talk about the properties of water as we go through the semester, but one of the things that I want to talk about is thermoregulation. So just again, just to kind of bring you back to that hydrophilic, hydrophobic nature, dispersion versus just dissolving. You can have non-electrolytes, things that dissolve in water but don't break down to give you ions, right? So glucose is an example. Why does it dissolve? Because it has all of these polar bonds, right? Anytime you see oxygens, you're going to have some polar bonds there, or nitrogens, you're going to have polar bonds there. So, it's dissolving in water, but notice it's not broken down to give you any ions. Acids, bases, and salts on the other end do give you that. Notice both of them are surrounded by water molecule. Anything that dissolves in water, what we're saying is that it is able to form polar bonds with water, and those polar bonds we call hydrogen bonds, okay? So you have this ability to form these hydrogen bonds with water, and as long as that ability is there, regardless of the nature of the chemical compound, it will be able to dissolve in water. And then there are compounds that are nonpolar. They do not have those, oxygens and nitrogens that give them that polar nature. So what do? They can't take part in this hydrogen bonding ritual, and they don't have a hydration shell surrounding them. So water, because the molecules tend to want to form at least 4 of these hydrogen bonds with other water molecules or with other compounds, they don't like it if it's less than 4. So what do? They kind of kick you out of the way, right? You're kind of preventing me from holding hands with my other neighbors. And because you can't hold hands with them either, we're just kind of exclude you. And depending upon the density of the compound, it either ends up at the surface or it ends up at the bottom, right? But hydrophobic compounds do not dissolve, non-polar compounds do not dissolve, right? Non-polar would be hydrophobic, and they end up adhering together either at surface or at the bottom, depending upon density. Now, what happens with amphipathic molecules? Molecules that have both hydrophilic and hydrophobic ends, right? So things like cholesterol, things like phospholipids, they have a portion that is hydrophilic and a portion that is hydrophobic. So anything like that, it needs to hide away its hydrophobic portion. It is water fearing. It doesn't like to be around water, so it needs to tuck it away somewhere where it isn't in direct contact with water. So amphipathic substances, if we were to consider phospholipids, for example, they tend to form two configurations. They either make a layer where, if you were to consider the head groups as hydrophilic and tails as hydrophobic, notice the tails are facing one another away from the water, right? And this is what gives plasma membrane its semi-permeable property, right? So if those hydrophobic tails weren't there, the plasma membrane wouldn't have that semi-permeable property. Anything that passes through this hydrophobic layer must itself also be hydrophobic or small enough. So we'll come back to that later on, but just think of amphipathic molecules as molecules that will on the surface, right, would be charged or hydrophilic, where they're interacting with water, but towards the core, they would sort of tuck away anything that fears water, non-polar parts of the molecule. So that's one sort of configuration they form. The other thing that phospholipids, just going back to that example, can form is a ball-like structure, a droplet, okay? If you've ever heard of LDLs and HDL and good cholesterol and bad cholesterol, you know, we're talking about these droplets, okay, where the hydrophobic tails want to be away from water, so they're kind of tucked in the middle, if you consider it kind of like a ball, they're towards the center of the ball, and the surface of the ball is made up of the hydrophilic portions. So, you know, this is a great way to carry hydrophobic nutrients in your body, right? So hydrophobic amino acids, or let's say not so much amino acids, but certainly hydrophobic vitamins, let's consider, or cholesterol, right? You know, you are going to have to somehow transport them through blood without actually getting the carrier protein. So how do you do that? The body tends to package cholesterol and all of these hydrophobic vitamins, such as vitamin A and vitamin D, in the core of these little droplets, right? So they're kind of protected by the phospholipids because the surface of the phospholipids allows this droplet to remain dissolved in water, and anything that is hydrophobic can be carried in the core very, very easily. Okay, so, you know, water mixtures, then what types are there, right? We just looked at, you know, what are the different types of different types of interactions between polar and non-polar compounds. So what kind of mixtures can there be within the body, right? And we'll use the example of blood, because that's the example your textbook uses. But, you know, there are, of course, fluids are found everywhere within your body, right? So depending upon the size of the particles that are suspended, okay, within a certain body fluid, you can have suspensions, colloids, or solutions, or emulsions, right? So let's walk through them a little bit, and let's use the example of blood. So blood is made up of blood cells. Leukocytes are white blood cells, or erythrocytes are red blood cells, and they're suspended in plasma, which is about 0.9% salt solution, right? So it's not just salt, there is all types of proteins and nutrients and things like that. But if you were to compare plasma to a solution, you know, its concentration equals that of 0.9% sodium chloride solution, right? So if you wanted to keep blood cells suspended within blood, blood must continue to flow, right? If it doesn't flow, it can you know, begin to separate components, which means that the red blood cells and white blood cells would separate out of solution, right? And you know, they tend to cling together, and they can form clot very, very easily. This is why it's important for us to, you know, get up and exercise a little bit if we've been sitting for a long time in one place, for example, on a plane, right? Because it's easy for your circulation to slow down, and, you know, clot formation can become much more easy. So suspension is, you know, large particle size, and generally, you know, you would have to continue to stir it somehow, right? Our circulation tends to help stir that blood, right? Cells through our blood. So think of it as sand in water. If you weren't going to continue to stir that sand, it would eventually settle out. So that's what happens with suspensions. In mixtures, so you think of just, you know, plasma, or think of cytoplasm of a cell. It's not just, you know, nutrients and electrolytes, it's also a lot of proteins, and it gives it this gelatinous appearance. If you've ever had Jell-O, Jell-O is basically collagen protein mixed together with water, right? So, and some sugar, of course. And so, you know, that's essentially the consistency, clot-like consistency that is of your plasma, and that is of your cytosol or cytoplasm, right? So, you know, it will change with temperature, okay? But that's essentially what a colloid would be. A solution would be, so if you were to remove not just red blood cells, but also the proteins from plasma, what's left is water with ions, such as sodium chloride and glucose, right? So it's very similar to what you would have in a cold drink, right? So you can take a cold drink like, you know, Pepsi Cola, and you can leave it for, you know, a whole decade, right, in your cupboard and come back and the nutrients would still be in solution. And the reason is because they're so small, once they're suspended, they remain suspended and they don't settle out due to force of gravity, right? So we have a lot of our nutrients in solution. They're not going to come out, right? They're going to continue to remain suspended. An emulsion is a mixture of water and oil. So in the human body, the example would be that of breast milk, which has a high fat content. So if breast milk was allowed to stand, it would separate the proteins that will generally mix together with the water, and the fatty compound would separate out at the top. Okay. So all of these types of solutions are found within human body. Now, why is, of course, so that's, what water is needed for, but, what about its other properties, especially in thermoregulation and so on. So our body is constantly using up nutrients, and as it uses up, nutrients that are found in our food, it not only uses some of the energy to fuel processes and make the proteins and other things that our body needs, but it also releases some of the energy of our diet as just simply heat. Okay, you exercise, your body is breaking down nutrients, and it can't trap all of that energy that's being released by the digestion of nutrients into ATP. So about, I believe, 70% of the energy released from nutrients is actually just released as heat, okay? And so your body has to continue to maintain the body temperature. Think about if every time we eat, right, the nutrients part, only 30% of the nutrients are used for other purposes, and the rest of the energy is lost as heat. So every time we eat, our body temperature must go up really high, but it doesn't, right? It remains at 98 point, 6 degrees Celsius, which isn't a standard anymore. But let's say it stays at 37 degrees Celsius, 98.6 Fahrenheit. But anyway, so at some particular temperature, which is unique for you, right, that is where the temperature stays. So what about our body helps us maintain that temperature? So you've already talked about, sweating as a mechanism to regulate body temperature. But what about sweating, right, maintains the body temperature, right? It is the evaporation of the sweat that helps to maintain body temperature. So what goes into all of that is just the understanding that water generally has a very high specific heat. So to raise the temperature of 1 gram of water by 1 degree Celsius, there's a lot of energy that is required. Yes, there are a couple of hands raised. So
why some people have trouble, like some people, they don't sweat a lot? Why some sweat a lot and others, even if they exercise, they have trouble sweating?
It has to do with your own sweat glands. Some people, the number of sweat glands are not equal in everybody, right? And so also if you, it also depends on your hydration level, right? And your electrolyte level, okay? So there are multiple reasons for that. You might have just genetic reasons why you don't, but if you have enough fluids in your body, you should be able to sweat a lot and cool down. If you don't have enough fluids, you're gonna heat up and not sweat a lot, okay?
Yeah, that can cause trouble, right?
Absolutely, because your body heat is gonna go up, and that's horrible for all the proteins and enzymes because they become denatured.
Okay, thank you.
Sure. So what you're seeing here is, I'm not interested in defining what that exact amount of heat is, but it's pretty high. And I want you to think about, water as we begin to heat it on stove to maybe make tea or coffee, right? So if you need to heat that water to 100 degrees Celsius, not Fahrenheit, it's 212 degrees Fahrenheit. You need to heat it really high to convert that water into its vapor form, right? So your body is able to use that property to cool down, right? So a lot of that heat that is being generated can be easily dissipated by allowing that heat to go towards breaking hydrogen bonds that hold water molecules together and causing them to go from their liquid state into their gaseous state, okay? So every water molecule has 4 hydrogen bonds, and don't think of them as permanent, right? They're constantly forming and reforming. So during your, exercise, initially you don't heat up very, very quickly. It's because of these water's heat capacity. It has an ability to absorb and retain heat. What does that mean? What it means is that initially as the body temperature begins to go up, right, the hydrogen bonds would be broken, but they would be immediately reformed, right? So all of that extra heat, that energy that is being released by your body will go towards breaking and reforming hydrogen bonds. Okay, so your body temperature doesn't really increase because, there are 70% of you is water. And, I can't even begin to count the number of hydrogen bonds there would be between those water molecules. And, your body temperature never really goes to 100 degrees Celsius, right? It's going to just fluctuate a small portion of 1 degree Celsius, right? A small percentage, about 20% within 1 degree Celsius. So that's enough to kind of, the hydrogen bonds are enough to kind of absorb that and keep your body heat at a particular level. Heat of vaporization, as I described. So, once you sweat, right, your skin becomes really warm because there's a lot more blood that is flowing to the surface of the skin, and the heat is going towards the sweat, the water within sweat. It breaks down the hydrogen bonds, and then it also causes them to enter the gaseous state so that they could evaporate, right? So it helps to cool down the body, right? Heat capacity tends to maintain the body temperature at 37 degrees Celsius, for example, and heat of vaporization helps to, get rid of that extra higher body temperature. So if it went from 37 to 37.2 degrees Celsius, so that 0.2 degrees Celsius would be taken care of by the heat of vaporization. So all three of them contribute to thermoregulation, but of course the heat of vaporization is directly involved because the water is being converted into vapor, and that takes the most energy. Okay, heat capacity is involved in maintaining the body temperature. And then the fourth property of water that comes to play is surface tension. So surface tension is basically the attraction for water molecules that happen to be at the surface. If you look at the this bug that is water spider, I believe, that's sitting on water, notice it seems as though water has a very thin skin on it. And so there isn't actually a skin on it. That property is referred to as surface tension. If you look at the water molecules that are on the surface, they are being pulled by water molecules from below. So there are 3 hydrogen bonds instead of 4 hydrogen bonds. And so they're pulled a lot more with a lot more force from the water molecules below. So it kind of gives it that skin-like appearance. Why is that important? Why is that surface tension important? The water molecules that happen to be on the surface, they're looking for an opportunity to form that 4th hydrogen bond, okay? That's their most stable configuration. That's what they want to be like. So they're looking for that ability. So if they come across another water molecule, they will form that hydrogen bond, right? And so one place where we have to sort of overcome that surface tension is every time we breathe in and out, the little bubbles called alveoli in our lungs, they tend to, blow up and then, constrict or expand, constrict, expand, and they go through that cycle. Every time they collapse or constrict, their walls come together, right? And on the surface of their walls, they have surface tension. And once they come together, the water molecules on the surface attract one another. So unless you had other ways of allowing that little bubble to blow up again, they will remain together and you would have difficulty breathing in, right? Every time you breathe in, they expand, right? You take in air. Every time you breathe out, they collapse to let out air, right? So if that surface tension has to be broken every time you are inhaling again. You have to allow the walls of the alveoli to expand again, and that can only happen in presence of lubricating fluids that have a lot of soap-like qualities to them. So we'll talk about that when you talk about the respiratory system. But the hydrogen bonding properties of water molecules are extremely important. Let's do a quick poll. Okay, let's do like 3 minutes. You're going to end in 10 seconds. Okay, guys, you're going to do this a little bit faster. Submit, please. I don't want you to lose the opportunity to submit your responses. Okay, all right, so let's share the results. Which of the following is true regarding a bond between nitrogen and hydrogen? Okay, it is a polar bond. It's not an ionic bond. So, there's certain things that I expect you to have remembered or just, read the chapter and kind of learn. But I also, I'm asking you these questions based off of what we just talked about. Is there a question on this? Okay, all right, so it is not an ionic bond, it's a covalent bond. It is a bond between an electronegative atom, nitrogen, and or which has greater electronegativity than hydrogen. So this is actually a polar bond. And in that polar bond, because nitrogen pulls the electrons with greater affinity, it's going to be, slightly negative. okay, in this case. Which of the following bonds is least polar? Okay, yes, so this is carbon and hydrogen bond is the least polar because I said that the two of them have about the same electronegativity. Which of the following will dissolve in serum? So again, NH3 would dissolve in serum because it's a polar molecule. O2 is a molecular oxygen, and anytime you have, oxygen is a sharing of electrons between the same types of atoms. There is no unequal sharing. With carbon dioxide, yes, there is, something called dipole moment and all of that I don't want to get into, but just know and memorize this for a fact that CO2 is also a non-polar molecule, which doesn't dissolve in water, and that I mentioned earlier on. And methane, because it's a hydrocarbon, it's a non-polar molecule because all the bonds are, covalent and equally shared. So NH3. What property of water is important for lubrication? So for lubrication, it's going to be, adhesion, because they are also sort of, attracted to the surface of the joint as well, right? So cohesion is just water's property to attract one another, right? So here we're looking at also their water's interaction with the joint surfaces as well. So it's going to be adhesion. What property of water allows you to maintain your body temperature so high specific heat? Something that I just went over with you guys, okay? All right, so let's talk about some of now the macromolecules, okay? Now, before we get into macromolecules, I'm gonna talk about pH. So if I don't get into macromolecules, I am spending some time just kind of doing some of these problems with you. So I'm gonna maybe share a video with you on macromolecules so you watch it and we kind of get over this. I spent a lot of time talking about all of this because it is so important to physiology, right? If you have no understanding of this or you don't know what's going on and why, when I'm talking about, muscle function or neuron function or bone function, you're not gonna understand it. So let's just spend the time now to understand it. Now, one of the things that is also extremely important is pH. And pH, simply put, is the hydrogen ion concentration in our body's fluids, right? So if we just take a simpler example where hydrogen ions are dissolved in water, generally these hydrogen ions would be contributed by an acid, right? An acid is a chemical compound that gives up a hydrogen ion, or what we also refer to as a proton. So, as you add acid to water, it contributes hydrogen ions. So it has a higher hydrogen ion concentration. And so it now has a lower pH, which I'm going to describe in a minute. And then you have a base, which when you add to water, right, it either sops up and absorbs hydrogen ions, and that's the definition we're gonna use, or it contributes hydroxyl ions, right? So in that case, the hydrogen ion concentration is low and pH tends to be higher, okay? The reason that there is this inverse relationship between hydrogen ion concentration and pH number is because of the formula that we use to calculate that pH. We generally use the log of the hydrogen ion concentration, and generally it's going to be very low compared to 1. Okay, so you don't have to know how to calculate and all of that, but just kind of understand the relationship between the hydrogen ion concentration and that of pH. Okay, the more hydrogen ions there are, the higher the hydrogen ion concentration and the lower the pH. Okay, and the reverse is also true, right? So you have fewer the hydrogen ion concentration, right? You're gonna have higher the pH. Okay, so I'm gonna talk about the range in a minute, but generally within water, right, the water molecule itself can break down into a hydrogen ion and a hydroxyl, right? So water itself is considered a weak acid and a base at the same time because it can either contribute hydrogen ions or hydroxyls. But because every time it breaks down, right, it produces equal amount of hydrogen ions and hydroxyls, it doesn't actually make the solution acidic or basic. The two will cancel each other out and it produces a neutral solution, right? So that equal number of positively and negatively charged ions would cancel out that. So when we look at the pH scale, and again, it's more important for you to interpret how to interpret this pH scale than how it's generated. But if we were to consider pure water that has those equal concentration of hydrogen ions and hydroxyl ions, we're looking at a hydrogen ion concentration of 1 in 10 million, okay, or 10 to the minus 7th. When you take a log of that, inverse log of that, I should say, you would get, and you use a calculator for that, you get a number that's called 7. Okay, so pure water is considered neutral because you have equal concentration of the two ions, and that's the pH of 7. Now, when you begin to go towards the smaller numbers, notice the scale ranges between 0 to 14. When you begin to go towards the smaller numbers, what we're saying is that the hydrogen ion concentration is increasing. So things are getting more acidic. So stomach juice has about the same hydrogen ion concentration as that of hydrochloric acid. So it has a hydrogen ion concentration of 1. Okay, in comparison, blood has hydrogen ion concentration of about 7.4, so it's slightly basic, right? And so as you move away from 7.4 to 14, things tend to get more alkaline or more basic. If you were to, compare the various pH conditions within our body, although cytosol and serum generally tend to be around 7.4, you can have other ranges of the pH. Like I mentioned, a stomach has a pH of, about 1. The fluids within small intestine have pH of about 8, okay, or initially 10 to kind of offset the acidity of the stomach, and urine has pH of about 8, right? So there's always a range. Do not consider that to be an absolute number. There's always a range, but so there are various pH levels that are found within the body, and they're important for the reason that pH is required for the normal functions of the body. Okay, so pH of 1 in the stomach is important because if you don't have that, you're unable to digest your foods. The pH of 10 is important, for example, in what's called duodenum, the first part of the small intestine, because that's where the highly acidic stomach juices enter the small intestine. We need to neutralize them, right? So that area is highly basic, right? And, if you think about enzymes that function in the stomach at low pH, at pH of 1, will not function at basic pH of 10. Okay, and so the body proteins are adapted to functioning at a specific pH, and that pH must be maintained. So pH is also a variable that is monitored by your body and is maintained via homeostatic mechanisms. And if you were to consider, comparing, acidic fluids with or acidic compounds with basic compound, acidic compounds tend to be sour if you were tasting them, and lemon juice, for example, would be acidic, and the basic and alkaline things would be slippery. Like soap is slippery because it's mostly alkaline, right? Now, one of the things that I want you to be able to do is to know that the difference between the numbers on the pH scale is a log difference. When we say a log difference, what it means is that the difference is 10 times. So from 0 to 1, it's 10 times. From 0 to 20, it's 100 times. From 0 to 4, it's going to be 10, 100, 1,000, and 10,000, right? So the difference between these pH value in hydrogen ion concentration is a magnitude is different by a magnitude of 10. Okay, so you should be able to interpret that. If I were-
Professor, I'm sorry to interrupt you. I just had a quick question. Is sodium bicarbonate a basic, and where does it fall along the pH value?
Not sure exactly, but I would assume it falls between 10 to 13.
Okay.
Yeah, it's very, very basic.
Okay.
Yeah.
Thank you.
Yeah. Okay, so you know, how is pH maintained within the body fluid? Let's look at the blood pH level. So normal pH, the range within which it fluctuates is 7.35 to 7.45, 7.4 being average. All right, so that is where your body is happy, the enzymes are functioning, the proteins are normal. And again, we're referring to serum, right? We're not just referring to the stomach's acidic environment. And so if the blood pH levels begin to deviate from this normal range and they go towards more smaller numbers on the pH scale, then we have a condition called acidosis. If they vary so that they're going towards the higher numbers on the pH scale, then we have a condition called alkalosis. pH imbalance it can be very, very deadly as well, because pH is extremely important to maintain the structure of the proteins and function of the protein. So regardless of which direction you move in, whether basic direction or the acidic direction, if it's outside of this normal range, it is eventually going to be deadly. All right, so it's tightly maintained within your body, and the organ systems that help to maintain that are your respiratory system and your excretory system, right? And the way blood pH is maintained is that there are what we refer to as buffers. Okay, buffers are these weak acid-base combinations that either give up protons when your body needs it, right? When would your body need it? When you are near that 7.45 range, we need to bring it down to 7.4, right? Or the conjugate base would accept protons if your body has too many of them. And when is that? When your body is nearing 7.35 range and it needs to be brought back to 7.4. Right. So yeah.
Question: what makes something a base or a weak acid?
What makes it a weak base and weak acid is that they don't immediately give up a proton or accept a proton, right? They generally are slow to do that, right? A strong acid would be HCl, for example, that gives up its proton very, very readily in solution. But a weak acid would be like the carbonic acid that I'm showing you here. It would do it at a very, very slow rate. Okay.
Thank you.
All right, so where is this carbonic acid coming from, which is going to act as that buffer that is going to accept the extra, protons or get rid of the extra protons, whatever it needs to do, and how does it form that conjugate base and so on. So the carbonic acid is continuously being produced by carbon dioxide in your body, right? So don't think of carbon dioxide as a completely waste product, right? It's not. It's still retained in your body to a high degree because it's used to produce a buffer that will help to maintain pH, and it's used for other purposes as well. You'll come back to it a lot. But so the way it's formed is that some of the carbon dioxide is found bound to your hemoglobin, and that's how it travels through your body. But 30% of carbon dioxide in your body will actually combine with water in red blood cells in presence of special enzymes, okay? In what we call carbonic anhydrase, and I think we'll come back to that again. But so the carbon dioxide will combine with water in red blood cells to make carbonic acid, right? So you can already see how it can form and reform and get, it can be removed from the body because it's a reversible reaction. The same enzyme that causes formation of carbonic acid is also the one that will reverse the reaction. Okay, now let's say that there was a lot of carbon dioxide, more than 30% that was retained, right? So there would be a lot more carbonic acid that would be produced, and because it's an acid, it will begin to give up its protons, right? So it will break down into a proton or a hydrogen ion and a bicarbonate base. Okay, so now you have these reversible reactions that can go in either direction. If CO2 would eventually be lost at the surface of the lungs, and if there is extra, bicarbonate ion, it will be lost via kidney. So it's lost in urine, right? So you can already see that what the CO2 is important, but in regulating the proton concentration, kidneys and lungs are important as well. So just to kind of help you see it, and of course, again, you're going to come back to it later on to talk about it in much more depth, but let's talk about what happens when so this is a natural reaction that is continuously occurring within your body within that 7.35 to 7.45 range, right? You either make more protons or you get rid of protons depending on which direction the reaction needs to move in. Let's say that the proton concentration is higher, right? So it can come from, say, diet. So you eat a lot of meat, and meats tend to be acidic and they tend to contribute, more protons, or you drink a lot of, acidic juices and things like that. But, there are other ways. And so let's say if the proton concentration is higher, right? What is that going to do to this whole reaction? It's going to obviously move in the other direction, right? If you have done chemistry, you know how bidirectional reactions occur. If you increase concentration of compounds on one side of the reaction, right, the reaction moves in the other direction, right? It makes no more sense to make protons when you already have a bunch of protons. So what are these protons going to do? They're going to combine with the bicarbonate ion that happens to be, naturally found within the body, and it will result in formation of carbonic acid, and carbonic acid would break down into water and CO2 would be lost at the surface of the lungs, right? Now, if you look at the bicarbonate ion concentration, right, it would go down, right? So you will have lesser of it within the kidney. So if somebody wanted to monitor if somebody was undergoing, alkalosis or acidosis, they just have to monitor the amount of CO2 that is being exhaled or the amount of, bicarbonate that is found in the urine. So you can imagine if the lung function is impaired, right, that extra CO2 can't be exhaled, and so that pH remains at the level that it is at. So extra protons can't be, removed from the body. Now, let's say the opposite happens. What if the proton concentration goes down, right? So you could be eating a lot of alkaline foods, a lot of vegetables and so on. They happen to be alkaline foods. So let's say, they are pushing your blood pH level near the 7.45 range, right? So how does your body react to that? If there are fewer protons in the circulation, then of course we need to make more protons, right? And the way if you're making more protons, then what that means is that you're making more of the carbonic acid, and carbonic acid is essentially being broken down into protons, and the bicarbonate protons would be retained because that is what's needed to bring the pH down within the normal range. And the bicarbonate ion would have to be removed, because if it's not removed, then it will just recombine with the proton to make carbonic acid again, right? So in this case, the bicarbonate ion would be removed via the kidneys, and the level of CO2 that is being produced would be lesser because, not as much of the reverse reaction is going on in this case. So you can imagine if somebody has an impaired kidney function, right, again, they're going to have issues with, retaining, maintaining their blood pH, because they're going to retain all of that bicarbonate, and whatever little amount of protons there are, they will combine together, and that will, continue to keep the blood pH towards the alkaline level. So look at that a little bit. I know it might not come to you immediately, but I will post these slides so that you have both of those images and look at it and sort of understand it. pH is also important in a phenomenon called ion trapping. And this is just an example just to give you why it's important outside of just, okay, it allows bodies, proteins to remain functional, and, it's needed to maintain the blood function and things like that. Another place where pH becomes really important is when we take drugs, right? And it's not just important for taking in drugs, but also excreting drugs in urine, okay? And so one example to give you is aspirin, okay? So all of you guys who are going to all of these healthcare fields, this is one of the things that you will talk about, anytime you take pharmacology, right? You take aspirin, and I don't care if you look at all of this complex structure of aspirin, but just focus on the carboxylic acid group that you have here, okay? And this is the part that is the acidic part, so it tends to donate a proton, okay? And like I mentioned earlier, weak acids, strong acids, they either readily give it up or they kind of wait around before they give it up. And oftentimes it depends on what we refer to as a pKa, which you don't have to worry about exactly what it is, but at certain pHs, they will give up their proton, at others they will not. So the way aspirin works is that at pH of 1, which is the stomach pH, it doesn't give up its proton, so it remains intact in its acidic form. It doesn't become ionic. But at the pH of 7 or 8, right, it does give up its proton and it becomes charged. Now, the importance of that concept is that if you've ever heard of drugs that get absorbed by your stomach's lining, aspirin happens to be one of those drugs that can be absorbed by your stomach's lining, right? So if you take it for pain relief, it works a lot faster than some of the other pain relief medications. So what causes it to be picked up very, very rapidly in the stomach is the fact that it doesn't become charged, right? It crosses the membrane very, very quickly. And once it crosses the membrane to reach your blood, it gives up its proton, it becomes charged, and then it can't cross the membrane because the charged ions and things can't cross the membrane very easily. So just to kind of give you a visual on that, here you have aspirin with its carboxylic acid on it. This is the stomach. Within the stomach, it would remain mainly in its intact form. In this form, it is able to enter cross the membrane and enter the circulation. Once within the circulation of pH 7.4, it dissociates, it becomes charged. And now if it tries to, re-enter the stomach, it can't do that because the only way it can re-enter if it was, not charged, right? So it helps to, the pH of the stomach helps to absorb drugs, and then prevent them from re-entering, the stomach's environment. So they remain suspended in serum. And then again, a similar phenomenon occurs when this drug arrives in the kidney. The kidney tubules help to expel it into the urine, and then, it changes its properties so that it can't be picked up again. So if the urine's pH wasn't maintained, you wouldn't be able to get rid of it via urine as well. So extremely important. Okay, let's do a poll and
Professor, can I ask you a question?
Of course.
So I have the slide that you posted on Blackboard here, and that last slide that you just had up, the pH ion trapping, the pH and ion trapping isn't on there.
Yeah, there are a few slides that are not there, so I'm gonna update those. No worries.
Okay, thank you.
Sure. So I'm gonna launch this poll. Let's take a couple of minutes. All right, so 15 seconds. Okay, I'm going to end the poll now. Okay, all right, let's share the results. All right, serum with pH of 7.2. Stop drawing on the screen, please. Thank you. Serum with pH of 7.2, okay, as opposed to 7.4 is lower in pH, so it's leaning towards acidosis, okay? For alkalosis, it would have to be like 7.6, let's say. Anything above 7.45 would be in alkalosis. What is the proton ion concentration difference between stomach's pH of 1? So this is where I told you that the difference between any two consecutive pHs is an order of magnitude difference. So, 10 times the difference from 0 to 1, another 10 times from 1 to 2, and you multiply the two values, okay? So it's gonna be 100,000 times, okay? All right. Then a person with a high serum pH, okay? If you have high pH, then you are going away from 7.45 to say 7.8, say, just to make it look a little bit easier, 8, 9, 10, right? And when you're going towards higher values of pH, you have fewer protons, okay? Fewer hydrogen ions, you have alkalosis, okay? Which homeostasis mechanism is at work in person with high pH, high serum pH? So they have a lot of protons, right? And if they want to get rid of that protons, they need to make more carbonic acid, and they need to get rid of more, carbon dioxide. And the only thing that makes sense here, actually there are multiple answers, right? So there's more carbon dioxide is produced and fewer protons are retained. Okay, yes, David.
For number 2, you said 100,000. Did you mean 1 million?
Oh, I'm sorry. Did I say 100,000? Yes, 1 million. Sorry. Thank you. Thank you for keeping that up.
For number 4, so there's 2 answers to that?
Yes. So so more CO2 is produced and fewer protons are retained.
Okay, thank you.
Okay, so I have literally 10 minutes left, and I think that I'm just going to talk about the organic molecules, and then the macromolecules, I'll post the video. You guys watch it on your own time, okay?
Professor, can you please explain number 3 again, please?
Sure. Where was number 3? Now I closed the poll already. Okay, here we go.
A person with high serum pH.
Yeah, so anytime we think high pH, right, we think fewer protons, okay? Fewer protons, okay? And more hydroxyls, if that was the choice. Fewer protons, more hydroxyl. As you increase pH, as you lower pH, it's, more protons and fewer hydroxyls, okay? Okay, all right, so let's just quickly talk about this. This would be the last topic, and then carbohydrates and all of that, I'll post a video on that. All right.
Professor,
yes.
just to ask you one last question. What was that formula again that you got the 1 million?
Oh, so anytime you're moving from one number to the next on the pH scale, it's a 10 times difference. So if you're moving from, 2 consecutive digits, it's 10 times the difference. If it's, from say 0 to 2, so that you're moving over a digit, it's 10 times, another 10 times, 100 times the difference, right? So if you did 4 digits, then it's 100,000 times the difference. But just keep it in mind that every time you are moving on the pH scale from one digit to next, that's a 10x difference. So we went from 0 to 7, I believe, or 1 to 7. Every time you move between these digits, you want to multiply by 10. Okay?
Okay, thank you.
All right. But obviously, the reverse would be true with the other side, okay? Because it will be decreasing in tenths. Okay, all right, so last thing that I'll talk about today, and then the rest would be in the video. Biological molecules, as I mentioned earlier, that we consider them organic molecules because they're made up of carbons and hydrogens. And we talked about carbon hydrogens being non-polar, right? We said that the electronegativity for carbon and hydrogen is about the same. So, you know, they aren't exactly going to dissolve very well in water. So if we were to just look at the bonds between hydrocarbons, you know, there is a huge variety of molecules that is possible because of the branching nature of the carbon atom, right? Because its ability to form 4 bonds allows it to bond with a whole bunch of different things, the other carbon atoms, oxygens, hydrogens, and nitrogens. And it can form chains, and it can form branch structures, and it can form linear things and so on. But none of them can dissolve in water. All of them are non-polar. So if you wanted to be able to dissolve them in water, because that is the solvent of our body, they must be associated with some polar you know, compound. So the way these hydrocarbons are made polar is by addition of what we refer to as functional groups. So here you're seeing a hydroxyl, and the hydrocarbon that we saw here, right, which is just made up of carbons and hydrogens called I don't think it's called benzene hexane. You know, it has no functional groups that make it polar. So no polar functional groups attached to it. If you were to dissolve it in water, it won't dissolve. It will kind of form a layer on the surface of water because it doesn't form hydrogen bonds with it. So if you want to form any hydrogen bonds and you take that same type of a structure and you attach to it some polar groups. So in this case, we have hydroxyl polar groups that will allow now a glucose molecule, it's no longer hexane, it will allow it to dissolve in water by forming hydrogen bonds with the water molecules surrounding it. Other polar functional groups can be carbonyl groups. We find them in proteins, for example. We find them in ATP because they're part of, you know, phosphates and things like that. There could be carboxylic acid groups that are found at the end of fatty acids, for example, also in amino acids that tend to make a hydrocarbon polar. So if we look at this molecule, this fatty acid, notice it's a large molecule and it has this large portion that is just hydrocarbon. So this is a classical example of an amphipathic molecule, right? And that this large molecule has a portion that is hydrophobic and another portion that is hydrophilic. And of course, similarly, there are other polar functional groups like an amine, which is NH2. When added to an amino acid, it makes it a polar compound. There are phosphates, there are sulfhydryl. You know, anytime you see oxygens and you see nitrogens and, you know, things like that, just immediately assume that they're part of a polar relationship and they will make this compound more polar. Now, how are these macromolecules held together, right? So, you know, one thing that we've discussed is hydrogen bonding, right? So that water will dissolve anything that is polar. So it could be polar amino acids, polar sugars, salts and acids and bases. Because of its hydrogen bonding ability, it dissolves all of them. But not everything is polar, right? There are things that were non-polar. So what holds them together is what we refer to as van der Waals interactions. So van der Waals interactions are kind of like polar. It's, if you were to consider oxygen, for example, right? Oxygen is, the molecular oxygen is O2, and there are 2 oxygen atoms that are doubly bonded together, right? And you know, although they're sharing electrons equally, they kind of, at times, electrons are just, you know, found around one atom more so than the other, okay? So overall, if you were to average out all of those events, the overall charge would be 0. There would be no slight positive, negative charge. But for that moment, right, when they are separate and they have that charge, that produces this attraction, okay, that allows things to, you know, remain together. So if you've ever thought about how cling wrap wraps around, you know, say a glass or a plate or whatever, it's the van der Waals forces. A lot of, you know, lizards that are able to rise and walk up the walls, they're also using these van der Waals forces to attach to the wall surface. So, it's also used by non-polar molecules to remain together. So it's a very momentary, brief separation of charge that keeps them, sticking together. And a third type of interaction is also what we've talked about is the hydrophobic interaction, right? Which we said is when hydrophobic compounds stick together because they're excluded by water, right? Water doesn't want them because they don't form hydrogen bonds. So in this example, we're seeing again a amphipathic molecule where purple region is the hydrophilic region and the orange region is the hydrophobic region. So several molecules that are similarly structured, right, will come together where they will interact with each other using their hydrophobic regions. Okay, reason is that this way they will kind of push it towards the core of this macromolecule that has formed at this point, and that will keep the hydrophobic region, as far away as possible, of course, from water. So although there is water around, but it's, there are regions that are not directly exposed to water. So this is referred to as hydrophobic interaction. So we're going to see that whether the attraction is between molecules, right, or within a molecule, like a large protein, right? These are the additional weak interactions that are going to hold these molecules together, right? So covalent bond, for example, can form the backbone of a protein, but it folds to acquire its three-dimensional shape via these weak bonds. Our DNA molecules are going to be held together via weak bonds, right? So just to remind you, right, the water is held, the molecules are held together in fluid form by these hydrogen bonds. And in order to break those hydrogen bonds, regardless of the amount of water you have, you need to increase the temperature to 100 degrees Celsius, right? So individually, these hydrogen bonds might not compare much to covalent bond and ionic bond interactions, but together, collectively, they are strong enough so that they hold our body's molecules together, okay? And so I think I'm gonna just stop at this, okay? And I will post the video for the rest. If there are any questions, please go ahead and ask, and I do have a couple of polls that I want you to be able to see and to get an idea of the types of questions that might be asked or how you need to be thinking about this information. So I will have those polls ready for you guys either at the beginning of lecture next week, or I'll post them on Blackboard for you guys to do on your own time, okay? There are a couple of people with raised hands. Alex.
Yes, I just wanted to know how to distinguish between van der Waals forces and London dispersion forces.
And which one, sorry?
London dispersion forces. I don't know the difference between that and van der Waals, the non-polar.
I'm not sure I'm getting London dispersion forces.
This is just something that I learned from organic chemistry, and I can't tell the difference between London and van der Waals. But if it doesn't apply to the course, then it doesn't matter.
No, I'm sorry, I wouldn't know. It's probably better that you ask your chemistry teacher. I'm obviously not a chemist, but
okay,
I'll look into it. Thank you for asking.
And then the quiz, where is that going to be posted?
It's going to be posted on Connect, and you will also have a link for it on Blackboard.
When is that due though?
I think by March, before the first exam.
Okay, thank you, Professor.
Sure. Yes, Sandra.
Yes, Professor. I see that some students were able to follow with the slides, but I didn't see where those slides were available.
These slides are still part of the first set of slides.
Oh, it's part of the first. Okay.
Yeah. But I will, like I said, there are a few slides that are unique to this, so I will update those and I'll post the slideshow.
Great, thank you.
You're welcome.
Bye-bye.
Hey, Professor, would you mind if
Yeah, no, go ahead, since you started talking.
Yeah, all right. So I only have one quick question before I leave. So what time will the quiz be posted today, or is it already posted?
It should become available automatically soon. Yeah, within next hour or so.
All right, thank you, Professor. Have a good day.
You too. Those of you who have no questions, you are free to leave, and I'll post anything you need to do on Blackboard. All right. All right, Fatima, you had a question. Fatima, are you there? Okay, Allison.
Hi, Professor.
Hi.
It's Fatima.
Oh, sorry. Yes. One of you. Fatima, you go ahead now.
Okay, I was wondering why you said specific heat was the answer to the question about body temperature being controlled. And you talked about how heat of vaporization helps to cool you down, but it wasn't the answer to the question. Can you explain that?
Which question was that? Sorry.
That was the second poll about specific heat being the phenomenon that helps in regulating body.
It was a specific question, right? So
Yes.
allows maintain body temperature at rest, right? So it can be heat of vaporization because you're not going to sweat at rest, right? That's when you're exercising. And cohesion is essentially just their attraction for one another, and adhesion is their attraction for other things, right? So such as the walls, the body wall, and things like that. So it was sort of like just asking you to really differentiate between specific heat and heat of vaporization. They're all related, right? You can't have one without the other, but I just wanted you to be able to think about it and say, okay, this is why.
Okay, so if cohesion was applied to like joints and everything, what would adhesion be?
Adhesion is applied to the fact that it adheres to the inside of the joint wall. Cohesion is their attraction for one another. So maybe it keeps the blood flowing because they are attracted to one another, right? This is the example where I gave you that if you were to, drop a drop of liquid nitrogen, it kind of dances around and disperses, but they don't stick together. But if you were to spill some water, they would eventually get together into a puddle. So that is the force of cohesion for water molecules, their attraction for one another.
Okay, yeah. Thank you.
Sure. Yes, Allison. Oh, you had another one?
Professor, I have a question. The quizzes that are posted on Blackboard for today.
Yes, so the quiz, the lecture assignments, whether reading or quiz, they will become available on a weekly basis, but they are really all due. They have a deadline of before lecture exam 1. So I want you to do them as you go so that you can retain this information before the exam, but I've given you the flexibility of doing it whenever you have time to do it. And they will become available, yes.
Okay, so basically you're posting these quizzes in order to us to learn to retain the information to memorize.
Exactly, they're very low stake testing. I wouldn't worry about them like it's an exam, but obviously I want you to read and take it seriously enough so that you are testing your knowledge and not just kind of quickly taking it to get 50 points or whatever, right? That's not the goal. The goal is to use it to retain the knowledge and ask questions if they come up as you're doing that.
Okay, thank you.
You're welcome.
Professor,
Yes.
are the quizzes timed?
Yeah, so the lecture quizzes they'll give you 60 minutes, an hour.
Okay, thank you, Professor.
You're welcome. Yes, Allison.
Hi, thank you. So can you point me towards like some resources? Because so I haven't taken chemistry in 5 years now. So I'm getting a little lost with distinguishing between polar and non-polar bonding and all of that I feel like is more in the chemistry world of things. Yeah. So if there's any resources you could point me towards.
I can post maybe a brief video, which I think is appropriate for your class.
Okay.
Or to differentiate between polar and non-polar compounds.
Okay, great. Yeah, thank you.
Absolutely.
And is that also something that because I'm taking chemistry now also, like intro chem, what specifically would if I wanted to ask my professor for help, would I just
ask the difference between polar and non-polar and what makes a polar bond versus what, I'm sure they would be covering this with you, shortly,
right
before the first exam, I'm sure of it.
Yeah. Okay. Thank you so much.
You're welcome. Yes, Vasi. Can I just call you Vasi, or you just tell me how to say your name?
I usually just go by Vicky.
Vicky? Okay. Awesome.
Yeah. I was just wondering for next class, I know we need to read chapter 3, but is it going to be chapter 3 and 4?
No, just chapter 3.
Okay, cool. Thank
Yeah.
you so much.
Absolutely. Yep, bye-bye. Andre?
Hey, Professor. Yeah, so this Monday in lab class, our teacher told us that if we don't do the assignments through Blackboard, the Blackboard links, then it's not going to be registered on Blackboard. Like when I did the assignments, I did them straight through Connect. So is there like going to be a problem there?
No. So that's exactly what he meant, but it just won't be recorded on Blackboard. It doesn't mean that we don't have evidence that you've done it. We can go to Connect and get it. It's just easier for all of us to see everything that you've done in one place, right? That's why Blackboard is important, because you will have your lecture grades and lab grades and everything. So if anybody wanted to see, they could see your Connect grades in that one convenient place as well. So if you click on the assignments that are on Blackboard, like there are links for Connect assignments given, then it will, I believe, automatically register into Blackboard as well. If they haven't, you don't need to worry. As long as your assignments are recorded as done on Connect, then that's all you really need to be thinking about.
Okay, thank you.
You're welcome. Autumn.
Hello. Yeah. I was just wondering, I know when we read the chapters, there's like questions that pop up.
Yeah.
So we have to do the chapter questions and then right away, like after, we should do the chapter quiz.
No. So you can take the quiz maybe, so if you've read the chapter and you've done the questions, so that's your reading assignment. The quiz, you can do it after you've like seen the lectures and talked about everything and you feel like you're comfortable taking it now.
All right, thank you.
You're welcome. Kayla.
Hi, Professor. So I have a question with the properties of water. How would, so why is like sweating less effective in cooling the body on a humid day?
On a humid day, the environment already is saturated with water, right? So it's not gonna, when we're sweating and we're evaporating, we're essentially asking to add more water to the environment, right? So it's not gonna happen because of that reason, that there's already a lot of water vapor in the air, say, and that prevents the addition of further water vapor. So you might be sweating, but it's not evaporating and it's not necessarily cooling you down.
Ah, okay, got it. Thank you.
Absolutely. Yes, Isra, you had a question?
Yes, I did. Hi, Professor. So for one of the polls, actually for the third one, I tried to submit like multiple times and it didn't submit, and then like the time was over. So I was just wondering if that was gonna like count against me. I think
I've noticed like you've been present, and so if you've contributed to others, I would see that. But you want to just do it just so that if I ever have to come back to it, and I'm not looking at it every day or anything, and it's okay if you miss a few here and there, I wouldn't worry about that.
Okay, thank you so much.
Sure. Yes, Connor.
Hi. I can't see the polls on my laptop for some reason.
So that's something that I unfortunately can't help with. Do you want to email help desk? They will send you some instructions to follow that'll help you figure that out. So email helpdesk@qc.cuny.edu and just say that you're unable to see the polls or you don't know where to look, and they will tell you that. Make sure.
Okay, I just wanted to let you know, because I don't want to like not get more participation.
Yeah, eventually that's what will happen if this continues, right? If I never see any polls from you, then I'm just gonna be like, okay, you just never get them, and I don't want you to, suffer. I don't want you to suffer because of that. So yeah.
Okay, thank you.
Yes, Fatima. Fatima.
Professor?
Yes.
Oh, sorry. I was just wondering, because I missed class last week because I had some problems with my account. I just wanted to know if there were some things that would help me catch up to.
So the lecture is posted on Blackboard. The recording is posted. If you look in the slide folder.
Okay.
Posted there, okay?
Okay, would you recommend any other thing to keep me like updated? Like
If you have any questions, come to my office hours and I'll be more than happy to answer any questions. As long as the lab is concerned, make sure that you speak to your lab instructor and make sure that you know, even if it's not, you're not able to make up assignments at this point, but at least do them so that you are aware what they, you know, required you to know.
Okay. Okay.
Right.
Thank you.
Sure. Hi, Shaya.
Hi. So last week you mentioned something about there being like an articulation agreement with Downstate for the nursing program. Can you tell me a little bit about?
Do you want to email me, because there are several components to it, and I will send you the whole thing, and then if you're interested, we can talk more about it.
Sure, I'll send you.
Yeah, and I will send you, you know, everything that you need to know, and then we'll go from there.
Thank you. And then I also had a question about the poll about the last question.
Huh?
When I was answering it, it's which homeostatic mechanism is at work in a person with high serum pH? So I was thinking like it's got like a high serum pH, so which homeostatic mechanism, as in like what would bring it down? But was the question, I guess the question was asking what was causing the high serum pH?
Right. So you're thinking, you know, is the respiratory system involved or the urinary system involved? Is that what you were thinking about?
So, as in, since it said like which homeostatic mechanism is at work, I assume that meant like to bring it back to a low like pH.
Right.
But was it asking like what was causing the?
No. It was asking what would bring it back, right? Homeostatic means to bring it back into range, right? So
then how would retaining fewer like hydrogen ions cause it to go back to a lower?
Oh, I'm sorry. Yes, you're absolutely right. You're absolutely correct. Okay, I got sort of, I didn't think about that in the moment. You're absolutely correct about that. It doesn't. It doesn't. I wish you had spoken up then, but that's okay. Thank you so much for speaking up now. It was the end of class and I was trying to rush through it, but you're absolutely correct.
So that.
Yeah, so it's really, okay, so so somebody who has high pH, right? They don't want to produce more CO2, and they don't want to retain fewer protons, right? They would produce more HCO3, right? That is the response to that. Thank you so much for pointing it out. I must have like just read the question just really quickly and not realized it. But if you are, you know, if you could point this out again next week, so that I can go over that. I am so sorry. I didn't read the question carefully myself. All right, Shakwana.
Hi, Professor.
Hi.
I just had a quick concern. So yesterday during lab, I didn't do the quiz because I submitted by accident because it was like mixed with one of the activities. So I emailed my professor about it, and there was only one attempt. So I emailed him and I wanted to see if, you know, there was a way I could you could reopen it by any chance, because he said that he couldn't do it.
Which quiz is that?
It was an APR for lab yesterday. There were about 4 assignments, so it was the first one.
Maybe 2 attempts for that. You should be able to take another attempt.
There was only one, so as soon as I hit submit, I didn't get to like even start the quiz, so I don't know if that affected it, and it's locked now.
Yeah. All right, email me and then I'll go and check it out. What's going on?
Okay, thank you.
You're welcome. Okay, Kayla again.
Hi, Professor. So I have a question with, I just forgot the question. You mentioned sulfide in the beginning of the lecture. I'm just looking at your, because the slides weren't posted, I'm just looking at your screen right now. So I was just wondering if you can give us more examples with the sulfide element,
So
I
if
guess.
yeah, so if we go back to the periodic table, it's one of those things that if you just commit to memory, it would be easier because it's obviously not a chemistry course and I'm not going to give you the periodic table. But if you look at this slide, for example, right, oxygen and sulfur are in the same period, and we know that you know, as you go from bottom, look at what's telling you that increasing electronegativity as you go from bottom top, and then again when you go left, right, it's increasing electronegativity. So not, you know, we're seeing more electronegative compounds. So you between all of these that are shown, just chloride is the most electronegative, right? And then you have you know, sulfur, and then you have, you know, oxygen and things like that. The ones that are part of the macromolecules, generally you're going to have phosphorus, nitrogen, oxygen, sulfur. Okay, if you think of any of these, just know that they are going to be part of polar molecules. I'm never going to ask you to compare phosphorus and sulfur, although you can if you wanted to. Like I said while I was talking about it, if you wanted to, you could, but I'm not asking you to compare them. The only thing that I'm going to ask you to compare them is with either carbon or with hydrogen. So all four of these, notice, are more electronegative than carbon and hydrogen. Okay, all right, so they're always going to be part of some sort of a polar bond.
Okay. Okay. And we were talking about electrolytes, and with the plasma membrane, we had an illustration up with some neons there. What is the significance of the neon K plus, Na plus, and Cl minus with the outer? Yeah. I'm just like a very visual learner, so it helps me to know the key.
Yeah, no, absolutely, I get it. I don't think I meant anything by that. Maybe it was just accidentally done.
Okay.
Yeah, no, nothing really. Thanks for pointing that out. I'll correct that.
Okay. And then the last question I have is with, can you move through the slides to the left? Yeah, right.
Keep on going.
Yes. What was the question again? Solvent. Oh, this one, here we go. So the nonpolar, so the hydrophobic substances that do not dissolve in water, the B, figure B, what would that be an example of in the body?
So, like I said, you know,
like, would it be like plaque buildup, or?
That's essentially it would become, right? So any fat that remains in your circulation, right, separate out, right? It's not going to remain dissolved. So either fat is picked up by your cells and stored as triglycerides, but you know, there is only so much room that your fat cells.
Got it.
So anything extra that remains in your circulation would kind of separate out, and then it would kind of become, you know, form a coating on the inside of your blood vessels, leading to plaque. Yes.
Okay, great. Thank you.
Oh, you're welcome. Yes, Alex, you had another question?
Oh yeah, sorry, last question. Just want to know if this course is okay for PA, physician assistant, because I saw in the Blackboard that there was something with Rutgers University, and so I didn't know if that promotion was trying to say that it's okay for this to be taken for PA
Right.
or
So
not.
anybody who so the biggest issue with PA schools right now is that so a lot of the schools are, you know, making a transition from non-majors level to majors level courses. And the reason I think is because there is such an interest in PA, right? The projected job growth rate for PAs is 30%. Okay, for NPs, it's 52%, right? So, and that's for next 10 years. So there is this explosive interest in PA schools. And so I believe they're trying to kind of regulate the process a little bit more so they know, you know, which students to pick and which students to not to pick. I know that there's some schools such as Hofstra and SUNY Downstate, and I believe to some degree St. John's and other schools that are beginning to say that they will accept majors level courses. But to be very honest, when they come in and they talk to the students, right, they either are being, politically correct. They're just kind of saying, well, we accept anything, and we accept students from community colleges too, and so on. So the way I look at it is that if you were to come to me and say that I don't know what school I'm going to apply to, I would say take majors level A&P, right? Because that will be accepted by everybody across the board.
Okay, yeah, that does make sense. Then I guess my only question then would be, will I have to retake this then? Because to be honest with you, I'm a bio major here, and this course is actually a lot more work than most of my upper level bio courses.
Are you kidding? It's because we're trying to kind of break down things for the students. For you, it's probably a lot of what we've already done, just very intuitive to you. If you have taken
this is a lot of this is reviewed, to be honest.
Yeah,
but that's the thing.
Lectures should just be reviewed to you.
Oh, it is. But I just think that in terms of like overall workload and what they're requiring, I actually think that this course definitely definitely got more work than some at least 200 level courses. So that's what, so they're gonna see that it says bio 40 and it's at a 100 level. So that means it's not major. So they'll think it's a remedial course. Is that correct?
No, I don't think they will think that. So what they will, so when you talk, I want you to come to the PA clubs meetings, right? And then talk about it. What I notice is that they're looking at an applicant at a holistic level. They're not going to pick and choose like, oh, he took bio 40, and although you've taken everything else at majors level, that's going to be
because I took biochemistry, like I took bio 371 here, and then the only thing that I didn't have on my plate was, if I'm being completely honest with you, I didn't want to take A&P 326 or 325 because Vesinin doesn't ask multiple choice questions, or at least not a lot. There's a lot of essays, and that's not my strong suit. So I wanted to take.
Actually doing everything multiple choice right now because it's all online. But regardless,
he's doing it all multiple. He's not asking for essay written answers.
No, he's not. That is for his. I think he's doing it for his other classes, but not for 325, 326.
Okay, I have to recheck. I have a friend in the course right now. I can flip over obviously at this point, but I might decide to take then at least 326 with it, but or 325. Thank you for the information. I'll come to the meetings. When is the next PA meeting?
I will post information on Blackboard, but email paprepclub@gmail.com and ask them to put you on their email list so you'll receive the notifications.
Okay, because I just don't want to be disqualified from like Hofstra right off the bat. Like they'll see that and they'll just disqualify.
Yeah, so from what I've spoken to the PA schools, I don't think just taking one course disqualifies you at all. But then, if you want to wait another semester and take 325, 326, that's fine too. Since you've already paid for this, if you want, you can stay and kind of build that foundation that helps you do really well.
That worries me just a little bit, Professor, because if you're saying that I should be taking it over again, you know what I mean, then I'm a little concerned. Do you think it's not worth taking?
It's because you want to take it. No, so this is what I suggest to you. Have you decided what schools you're applying to?
I was going to apply across the board to like 20 different schools. I really don't care where I go, just want to get in.
Applying across the board to 20 different schools, I'll be honest with you, there are plenty of people who've taken Bio 4041, applied to PA schools and gotten in. If you were to come to me before this and knowing that you're a bio major, I won't recommend this course for you. Right now, all the Connect assignments and all of that we're doing, it's really for the students who are at non-majors level and they need that extra help, all that practice and so on to retain that information. Somebody like you doesn't need it. A lot of this, like you already know, Vesinin isn't going to start at, diffusion, osmosis, macromolecules at all with you. He's going to straight get into I believe integumentary system and then skeletal system and so on. So, there's certain level of information that we already expect of you. So, it's up to you. But if you are applying across, the board where you're applying to 20, 30 schools and you have taken everything else at majors level, if you do well in this course, I can recommend you to Dr. Vessanen so that you can take 326 at a majors level, but you have to do well, right? I mean, and by well, I don't mean like you have to be A+, but if you're interested in taking 326, I can certainly recommend you for that. But you know, it's your choice, of course, you know, whether you want to stay, drop, repeat, whatever. I just want you to know that there is no, you know, hard and, you know, fast rule that says that you cannot take non-majors level or you should only take majors level.
Okay, thank you, Professor.
You're welcome. Any other questions? All right, guys, you guys are free to leave, and I'll, if anybody has questions, I'll be there in my office hours.